starting with Simon Pierce, who is Saving the Whale Shark, Global Research and Conservation Priorities for the World's Greatest Fish.
Geez, you can see the curvature of the Earth from up here. Just waiting for my talk to come up. Cool. Fantastic. Well, hopefully you've all had opportunities to familiarise yourself with whale sharks. Otherwise, there is plenty more opportunity to improve your life choices this week, so that's great. I wanted to talk about whale shark conservation. So we updated the red list for whale sharks last year. Sadly, they are still an endangered species following the previous assessment in 2016, and the population is still actually decreasing as well. And the kind of headline result from that is that there's been more than a 50% decline over the past 3 generations, but particularly since the 1980s. And in the 1980s was the start of some large commercial fisheries for whale sharks in the Indo-Pacific. Sadly, those fisheries pretty much ran out of whale sharks to catch quite quickly. There was also some amazing conservation efforts to close those fisheries down. But we're in a situation now where we've got a heavily reduced population of whale sharks, and we've still got quite a few active and emerging threats, which is either causing continuing decline or slowing down the recovery. Now, whale sharks are a widely distributed species. They just like seawater over about 22 degrees Celsius. But there's been several genetic studies done on the species now, and there's been a pretty consistent demarcation between the Atlantic and Indo-Pacific populations. So for the red list, we were able to subdivide the subpopulation assessments, and looking first at the Atlantic, we estimate based on habitat modelling and effective population size and a fair bit of guesswork that there's about 25% of the global population in the Atlantic. And one of the issues we've got over there is that we haven't got much in the way of trend information. So we've got some shorter term stuff, we've got some unpublished stuff, but the only kind of long-term, broad-scale time series is from the Eastern Atlantic from tuna fisheries observers. So it's hard to get a handle on what's actually going on with the whale shark populations over there. But in terms of threats, we do know there's a big risk of ship strikes. So there's large whale shark populations, aggregations feeding on the surface in the Gulf of Mexico and off the Yucatan Peninsula, and that's very close to some large shipping routes. So we see a lot of ship strikes on whale sharks, and that's kind of the core conservation threat that we know about as well. And then we've just got to learn more about what's going on in the Atlantic. So switching over to the Indo-Pacific, where the other 75% or so of the whale sharks live, that's where the large target commercial fisheries have taken place. So we estimate there's been over a 65% decline in whale shark populations over that area. The problem is that it's really big. So we kind of did a bit of a straw man assessment for the green status in 2021 to sort of, based on what we know about whale shark population connectivity and also like sort of reasonable management units, we kind of strawmanned it into 4 different spatial units for assessment. So starting with the Western Indian Ocean, we have some of the better trend data from that region. We've got broad-scale Mozambique Channel data from tuna fisheries observers, which has shown a steep decline in whale shark sightings to the mid-2000s. And that's about when we started work in the Inhambane province of Mozambique. And we've seen an 89% decline in sightings over there, and also a 99% decline in the number of large 7-metre plus whale sharks that are being seen. So it's been a really steep decline. And the main conservation threat we've been able to identify over there is accidental catch in particularly coastal gill nets. So that's what we're trying to work on now. But we really need to get a better understand of like what are the drivers of such a substantial decline, because it's not super obvious to us at the moment. So we're working on what we can see, and we're trying to identify what else might be causing that decline. Moving north to the Arabian Sea, again, we have a real shortage of long-term population trend from that information. There was a large fishery for whale sharks in Western India, and then there's sort of shorter-term stuff from other locations. But The Arabian Sea is a particularly high threat environment for whale sharks. Again, we have a big problem with ship strike risk over there. Some of David Robinson's work of Qatar up until 2016 found 43% of the sharks there had significant scarring, a lot of which were really large propeller wounds from like large tankers and things. There's also a really big offshore tuna gillnet fishery in the region. So there's some of these nets are up to about 30 kilometers long. They're absolutely enormous. And since the inception of that fishery, it's estimated to have caught over 4 million cetaceans. So we don't know too much about its effects on whale sharks, but we suspect it's going to be significant as well. Moving across the Asia Pacific, that's where a lot of the large fisheries for whale sharks have taken place in southern China, Taiwan, and the Philippines. And I think we've got a real shifting baselines problem over here, in that most of the trend information we have is from quite recent, so we haven't necessarily captured that steep decline from fisheries. We have got a broad-scale tuna observer data set, again, from the Western Central Pacific, which has shown a broad decline through the Western Central Pacific. And this is also a high threat environment. We really need to quantify just how many sharks are still getting caught. There was a really good interview-based study done off the southern Chinese coast that was published in 2023, and they estimated there was still 540 whale sharks getting caught annually along that coast. And we're talking about a species where the largest aggregations we know about are maybe just over 2,000 individuals. So it's a pretty substantial number for whale sharks. And also, in terms of those offshore tuna net fisheries again, we've got over 280,000 gillnet boats operating out of Indonesia alone, which also probably affecting whale sharks quite a lot. So finally, across the Eastern Pacific, one of the kind of open questions about this sort of region is how much it's been affected by those big fisheries elsewhere in the Pacific Ocean. There has been some broad-scale declines, again from tuna observer data, and then there's sort of shorter-term sighting data. So in terms of threats over there, we do know that there's large gillnet fisheries off Peru. There's over 100,000 kilometres of gillnets being set each year over there. They've got a high catch of mobulids, and there's also a lot of entanglement with cetaceans, so again, probably affecting whale sharks. Cool. So I wanted to sort of summarise, basically we know a lot about juvenile male-dominated feeding aggregations for whale sharks, and the less positive way to say that would be we don't really know where the babies are, we don't really know where the adults are, and we don't actually really know where the females are. So there are still quite a few open questions. So if you're interested in whale shark research, I would highly encourage you to get on board because we've got lots to do. We really need more long-term trend information, and particularly trying to get some of this broader scale stuff. So whale sharks are highly mobile, and it's really hard for us to ascertain, like if we're seeing an increase or decrease at a particular site, how that might relate to environmental change or threats over a broader scale to see how that's affecting the broader population. And yeah, I'll just end with a bit of a shout out to sharkbook.ai, which is the photo identification library that most of us are using for sharing our collaborative photo identification data sets, which really enables these broad-scale population assessments. So we really need more people publishing long-term trend information. So if you have got whale shark photos, or if you're whale shark photo adjacent, we'd love to get you on board with this big open source collaborative effort. And whale shark conservation is a pretty big topic, so if you'd like to download the slides and have a bit more, you can just use this QR code as well. Thanks very much.
Awesome way to kick this off, and we have time for questions. Anyone?
Hi Simon, great talk. I just wondered, with the increasing trends, I wondered if it accounted for survey effort, because I know there's a lot more kind of in-water whale shark IDing happening in some areas, and if that kind of has also contributed to what we know in certain regions.
Sorry, in-water survey effort?
Yeah, so like the survey effort about how many whale sharks people are actually seeing, like how much time we're spending in the sites, is that also part of the increase we're seeing in some of these areas?
Yeah, I think so. It's like a lot of the trends we had for the red list assessment were based on like Freya Womersley's paper with the expert surveys. So it's partly kind of vibes based, and like really there's not a long time series in some of these places. So given that there has been a lot more increased survey effort in some locations, and more awareness amongst like citizen science and dive industry and that, yeah, it's pretty hard to disentangle. So yeah, if you could rephrase your question in the form of an answer, Sally, that would be fantastic. Yeah. Cheers. All right, thanks everyone.
All right, thank you, Simon. On reef shark habitat use.
Hi everyone, I hope you guys can hear me at the back. Yeah, thank you all for being here today. I'm presenting one of my PhD chapters that I've just finished, and it's about coral reef associated sharks and what happens to them as we start to lose our coral reefs due to climate change. So if you think about sharks and coral reefs, they've been around for hundreds of millions of years. However, we're losing our coral reefs very rapidly due to climate change. And with that, it's an issue where coral reef complexity is getting homogenized. That means they're basically starting to flatten, and we're starting to lose those pretty beautiful corals that we all love when we go snorkeling and diving. Now, shark movements, however, as you've just seen in that example, they're often investigated at hundreds of meters or kilometers of scale. So people look at these systems like our study system here, and they say, are they moving from the lagoon to the fore reef, into the channel, to another island? What I really wanted to capture, though, was the fine-scale complexity within the fore reef, so within the reef habitat. And what I found, and what I've looked up yesterday on Google Scholar again, is there's quite a little integration between fine-scale complexity and movement tracking. And obviously that leaves a bit of uncertainty as to when these coral reefs are disappearing, what happens to our sharks. So I wanted to combine what's already out there. So I know there's a lot of shark nerds in the room, so you've probably heard or have done acoustic telemetry tracking. But then I come from a coral nerdy background originally, so I wanted to combine it with what we call structure from motion photogrammetry. So I essentially had 3 simple questions. How does reef structure influence the habitat use of some common reef-associated species, so like the ones that you see here, so the blacktip, the gray reef, and the lemon shark? At which scales do we see a signal? So looking at centimeter scales, like sub-meter scales, very, very fine scales. And are some species maybe more resilient to reef homogenization than others? So I've had the pleasure to do my work in the Seychelles, and I've worked with the Save Our Seas Davos Research Center in this beautiful location that you can see here. And we've tracked 65 different sharks over 2 years, and we had over 1 million detections, so quite a bit of data to sort through. And we've used a subset of the receiver array. So we excluded the receivers from the lagoon, we excluded the ones offshore, we just wanted to see what is going on around the reef. As you can see the species here, if you've never seen them before. And yeah, just to give an example, for our gray reef sharks, for example, they were quite concentrated, around the channel and at northeastern point of Davos. So didn't really reinvent the wheel when we looked at that, and we're like, okay, gray reef sharks like channel habitats, shock. Now, with that, we then obviously have to collect our photogrammetry data. So you can see Amy in the background here. It's quite simple. You essentially swim over the reef. You have a GoPro camera. It's as simple as that. And you take continuous pictures of the coral reef underneath you. Jumps a little bit, but I hope you got that. So we did that around each acoustic receiver site. We did 3 10 by 10 meter quadrats, and in total, we've mapped 6.5 square kilometers of reef. Then we created these models at 1 millimeter resolution, so very, very fine scale. The blue squares are each individual photo taken down from the top. You can see then a program stitches these together. It looks for points that overlap and then starts to build that 3D model. And it's basically an iterative process where your computer starts smoking and you have to let it run for a couple of days and then you get there eventually. And yeah, it is a bit complex, so obviously we can talk about that more later, but that's essentially what it looks like in a nutshell. With that, we looked at 4 very common metrics. But before I get into that, we essentially did these quadrats in the receiver's detection range. So our receivers had a detection range of around 160 meters, where we can detect the sharks. So we then said, okay, let's do our reef surveys within that range, do them randomly, and see what our receiver habitat actually looks like. The metrics that you can see on the left, they're essentially proxies for different complexity, different coral reef health things. So roughness looks at seafloor habitat variation. How wavy is it? How up and down does it go? Slope is obviously looking at hydrodynamics a little bit more. TRI and VRM, they're measures of seabed variation, and they're quite relevant for mapping. So they're proxies for heterogeneity and structural complexity as well. We've modeled them at 5, 10, 25, and 50 centimeters within our quadrats. And essentially, I picked them at random because I was like, I have no idea where we're going to see a signal, so let's just roll with it and see what happens. We use two different things. We've looked at the mean complexity per receiver site. So we've combined all of them and said how complex is our site. But then we also did a little bit of maths and looked at the deviation between the different quadrats to just capture a little bit more of that complexity at each site. So here's where the fun part starts. We did two models, and at first we wanted to see, okay, where do we see signals? What scale actually matters? Because all of them were super, super, super correlated at these fine scales. So I took my photogrammetry metrics, took the different scales, and essentially just put them together with our shark detections in our residency and said, okay, at which scale do we see a signal? Which ones should we investigate further for the different species? Once we've had those intricate scales and metrics, we then again said, okay, our detections and residency. So detections is how often did the sharks ping at the receiver over the study period. And then the residency is basically an index of how many times how much time do they spend there per month that we surveyed. So then we said, okay, we saw a signal, for example, at roughness at the 5 centimeter scale. And then we threw in some random effects. So we throw in time, so the time of year in terms of months. We looked at depth, so how deep was the receiver. And also receiver identity, right? Where was the receiver as well? Because that, as you saw for the gray reef sharks, it will matter. So what we found was actually quite surprising, but also not very surprising. In the case of the blacktip reef shark, we found a very, very strong positive effect and relationship between blacktips and positive metrics of coral reef complexity. So almost 70% of their space use and habitat use was defined by different complexity metrics at different scales, both at the mean and at the standard deviation side. So they just love complex reefs, which obviously you guys have probably seen blacktips. I mean, expected, right? However, here's, I think, where the interesting part comes, and that is the gray reef sharks and the lemon sharks, they did not really care. We couldn't find any significant responses to what we've tested here. The gray reefs, in this example here, there was a negative relationship with one of the metrics. Some of them were positive. All of them were actually not really telling us very much. For the lemons, we saw a little bit of a seasonal pattern that popped up, but that model there explained like 1% of their habitat use. So for them, they don't really care about complexity. Now, what does that tell us when we look at why is that conservation relevant, and what do I want to take, or what do you guys should take away from here today? I think some species are going to be more vulnerable to climate-induced habitat change than others, but I think there is a reason for hope. So the blacktip reef sharks, they're going to struggle, especially as they're being fished and they're losing their habitats. But the other species, I think they may be quite resilient. If we just leave them alone and maybe stop fishing them, I think they can hopefully cope with some of that change. Doing some little simple maps can pick up patterns where your resources are going to be limited. If we would have mapped the entire detection range of each receiver, I would still be sitting at my computer running those models today. And in future, which one of my examiners is here, so he brought that obviously up, we need to think about other factors, right? How is prey going to factor into this? Temperature, location in the world, right? Is it going to be the same in the Maldives or in the Pacific? And then also expand this to other reef-associated shark species, right? There's so many more species we can do this with. So yeah, if you guys have an array and want to do some of this stuff, then let's have a chat, basically. Yeah, and with that, I just want to say a very big thank you to my founders and my partners as part of the last couple of years of my PhD. Thank you to the Save Our Seas Foundation for funding this, and the team at the Davos Research Center for having me and for helping me with all of this. And yeah, you can obviously go and send me an email, send me a carrier pigeon, or just come and talk to me, and I'm happy to take some questions if there's time. Thank you very much.
Thank you, Nico. We do have time for one quick question. Sammy.
Hi, hello. more complex reef will be harder to detect a shark potentially. So is receiver detector range variable between the different complexities of a reef?
Yeah, absolutely. So if you guys caught that, Sammy was asking if complexity will factor into our detection range and where we can detect these sharks. So we took a quite conservative approach here, and we stayed quite close around the receiver, to pick up those patterns. But what I would like to do in the future is to do more range testing and to really say, for these complex receivers, they're gonna have a smaller range, right? Like there's so much noise going on, so it's probably gonna be more concentrated around the receiver. And then for the ones in more open areas, they're probably gonna be quite far away, these animals. So maybe we should also survey accordingly. So that was just the first step, which hopefully, helps to solve those questions in the future and when we incorporate that then. Thank you for your question.
Thanks, Nico.
Thank you.
All right. Next up, we have Sergio Madrigal-Mora, speaking of drivers of white shark movements and residency at the Neptune Islands Group, South Australia.
Hello, everyone. Thank you for coming, and I'm very excited to show you some of the results that I have so far for my PhD. So to understand and truly understand animals ecology, of course, we have to understand the movements, and we've seen some great examples of this. As we've seen from the other presentations, some of the broad drivers that influence shark movements may be feeding, for example, some suitable habitat, reproduction as well. And a lot of the time, this leads to sharks going to the same places and forming aggregations. A very interesting example of this is the white shark, where we have two extremes, right? We have a shark that moves around a lot, broad-scale movements across their whole distribution, but then they also have specific aggregation sites where they spend prolonged periods of high residency there. So for example, in Australia, we have white sharks throughout the west coast, south coast, and the east coast. But throughout that big distribution, they're aggregating in some sites, and we're going to focus in one of them, which is right here in South Australia, and that is the Neptune Islands. The Neptune Islands are an island group composed of two main subgroups, which are the North Neptunes and the South Neptune Islands. And of course, they support a very diverse ecosystem, and part of that ecosystem is a big abundance of white sharks. Because of the frequency that white sharks are seen, this place is one of a world-class cage diving site and the only active site in Australia. So we're really interested in understanding why white sharks are present in the Neptune Islands. And we're looking at this at various scales. So first of all, for our first study, we looked at the residency of sharks throughout the Neptune Islands as a whole. And then we went and took a closer look at each of the islands individually to see how the use differs between islands and how they transition between islands. So for this, we used the beautiful acoustic telemetry we've all been talking about. And we have a data set starting in 2013 all the way to present. So far, we got 216 sharks. They're mostly males, but there's still some females that come up. And they range from 2 meters to around 4.5. So as a measure of residency, we use presence and absence per month and put that into a GAM. And we tested as many variables as we had available, some of them environmental, others representing prey availability, and then of course cage diving, as cage diving uses a burley and bait to attract sharks closer. But we found none of them, including cage diver, had an influence on the monthly residency or presence-absence of the sharks. We wanted to test for pinnipeds, of course, which are present in the islands, but the surveys there stopped in 2013, right as we started our study, so the data is not available. However, we did find one variable that was significant, which was seasonality, and it had an interaction with sex and size of the sharks. So what we see is the males are present in the spring, while females are more likely to be there in the autumn and winter, especially the large females were more likely during that time. However, all of this seasonality is not correlated with any of the environmental parameters we have, so we're still left with a bit of doubts. And then, of course, we found a very high influence of individual variation, meaning that even though we have these trends, they represent the trends for the population, but a lot of individuals might not follow them at all. We tried to do another similar model, this time with visit duration, meaning how much days each shark stayed when they got to the island. And again, we found nothing really influenced it very well. And we found a very high individual variation as well. So we wanted to take a closer look at the moments where we had more extreme conditions of environmental variables. And we identified some extreme events, 4 extreme events. At the top, one with strong El Nino conditions. Then we also analyzed 2 very cold autumns, and then 1 strong upwelling event. And we looked at the environmental variables before, during the event, and then after. We then used a hidden Markov model to see whether sharks were at high or low presence states, and tested whether the high or low presence of sharks occurred in extreme or normal conditions. This is a bit of what we found, which again, not a lot of influence from the environment. As you can see in this plot, we have in the rows each of the events, and in the columns the environmental variables that were present at the events before and after. So take a closer look, we can see the El Nino conditions. We had high presence at the event that had really, really strong El Nino conditions. And then we also had high presence during the event that had very strong La Nina conditions, which are the complete opposite of oceanographic conditions. Then if we take a look at the temperature, we see that sharks were present and absent throughout the whole range of temperature, specifically looking at this case of the El Nino event. And then for chlorophyll, we see that during the strong upwelling, that point far off to the edge of the plot was the peak of the upwelling, and sharks were absent there, but also sharks were absent even when chlorophyll was a bit lower. So it seems like sharks are present or absent both when the conditions are extreme as well as when they're normal. So then move on to the second study, trying to look at a finer scale and see what more we could learn from these sharks. And again, like what we're looking at is the use of each island and the transitions of each island between the North and South Neptunes. So we used another hidden Markov model, this time to predict the probability of the sharks moving between islands or staying at islands. And we found that sharks tend to be temporary residents at the island that they arrive at. So when they arrive at an island, they're more likely to stay there than to move on to the next one. You can see that in the purple, really high values of probabilities for them to go from north to north, south to south, or just stay absent. And we see that, of course, this means transitions are quite infrequent. We average 2 transitions for every 2 weeks that a shark might be present in the island. And we see them, little animation. So these transitions can be as short as 3 hours, but that is very infrequent. They're often much, much longer, lasting around 20 hours on average, which kind of tells us that the sharks are not going directly in between islands and kind of taking meandering paths across them. And so if we have indirect travel and also infrequent transitions, this starts to hint at a possible central place foraging behavior, where a shark might arrive at one of the islands, stay there for rest and navigation, and forage in the nearby areas while returning. And once the shark has exploited the resource patch near there, they might move on to the next island, foraging throughout the path to get there and repeating such behavior. Interestingly enough, we did find some differences in use. The sharks seem to prefer North Neptune over South Neptune. And we think that is because there is a greater abundance of pinnipeds in North Neptune than in South. But again, this is based on the old surveys from 2013, so we must assume that this still continues so many years after. So what about other factors? Of course, cage diving is what comes to mind. And in fact, there is also a preference for the cage diving operators. They are active at North Neptune far more often than they are in South. And this is because North Neptune is closer to port, so it's much cheaper for them to go to North Neptune and stay there than to be moving down further to South. So this means lower costs. And if they have been active for so many decades there, is it possible that that is making the sharks prefer North Neptunes as well? We don't think so, because a lot of different studies from our lab have shown a very limited effect on sharks from cage diving attraction. And this is under current management regulations that are pretty effective. And also we find for our models that sharks do not transition between islands when the boats do, so they're not following the boats at all. So in summary, we find that of all the variables we tested, not much of them were affecting the shark movements, but individual variation had the strongest influence on what sharks would do. So the next steps is to look at these individuals and how each of them are behaving and see if there's any consistency in what they do to better understand how these sharks are moving. Thank you so much.
All right, thanks, Sergio. We have time for one really quick question if someone has right at the front.
Do you know much about the effects do you know much about the effects of the algal bloom that happened in South Australia last year?
Yeah, we've had the sharks absent throughout the time, but the algal bloom hasn't really come to the Neptune Islands and affected it directly. So what we think, and also well, seeing the strong upwelling event where we had super high chlorophyll concentrations and sharks left for the big peak, but still returned when concentration was still pretty high as well. So we would think that the algal bloom is not necessarily affecting them directly, but of course, you know, all the prey items that are being affected could possibly make them move a little more. That's also stuff we're going to look at with newer data further on. Thank you.
All right, guys, thanks for staying. Up next, we have me, Sam Sherman, talking about ecological niche partitioning. Yeah, so I'm really excited to be here. This is my first Sharks International as a PI, coming with students, which is really exciting, but also means I don't have heaps of my own work to present. So instead, I am introducing my lab, which is the MEME lab. It stands for Movement, Ecology, and Management of Elasmobranchs. And our work spans a lot of different areas. It was established about 2 years ago. We're currently housed at Deakin University, and we work on ecology in Southern Australia and Southeast Asia. We work on fisheries and international policy, including CITES, and all different scales of fisheries assessments and managements, artisanal, industrial, and international. And we also don't just do shark stuff. I have a bunch of students who aren't here who work on smaller fisheries in Victoria, bivalves, flatfish, or flatheads. I have some oceanography students and some that are working on technology. And so we do a lot of different things. You can check out on the website all the students that are currently there, and if you want to follow our adventures on Instagram. So one of our students, Tia, presented yesterday. Tia Burke is a PhD student working with Lauren Meyer at the Southern Shark Ecology Group in Flinders and Britt Finucci at Earth Sciences New Zealand. She just spent a month on a boat collecting all of her samples. You missed her talk, but that doesn't mean that you can't talk to her about all of her cool stuff and ask her to show you more pictures like that one. So make sure you find her if deep sea stuff is of interest. Christina Ekanayaka is here. She did a stock assessment with Blue Resources Trust, and she's speaking tomorrow at 2:35 in here, so make sure you come back. She did a mobular stock assessment on the Sri Lankan mobular fishery. Alex Montanaro has a poster today. He's working with Vinay Udyawer from Sharks Pacific and Hugh Peterson from Innovacy. And if you do acoustic telemetry, which if you're in this room, you might, definitely see his poster. He's gonna make your life way easier in the future, helping to create array optimization and customization. Darren Wong, who is not here, does a lot of age and growth stuff. If it interests you, you should definitely work with him. He's also worked on fisheries stock identification. Louisa Graff, who also has a poster she's presenting today, is doing movement of trophic ecology of southern fiddler rays. That's one of her rays, hanging out, spending a lot of time in Swan Bay. And that's where I'll tell you now about our big project that we've kind of started in Port Phillip Bay, which is a large bay that's almost 2,000 kilometers squared, but very shallow, less than 30 meters across the whole bay. It is less than 8 meters across 50% of the bay. It's located in Victoria in the south of Australia. And this is what it looks like zoomed in. So you can see Melbourne's at the top there. It's very, very busy. It's the biggest shipping port in Australia, and there's a very small opening, less than 4 kilometers across. Super strong tides, really shallow, lots of vessel activity. There is also a lot of recreational fishing and aquaculture in the bay, so heavily used. And our lab is right near the entrance of the bay, so we're in the perfect spot to look at movement of sharks in and out. So that's what we've done. Again, this is the bay. This is our current receiver array. Depending on where servicing is at, we have between 70 to 80 acoustic receivers across the bay. But you can see there's a high concentration at the heads, or the opening. And in that opening, we have a double curtain. So ideally, anything that's coming in or leaving will be picked up, and we can look at that use. And then we also have a really fine scale array in Swan Bay that Louise is really using for her fiddler rays because we think it might be a nursery area. So, like I mentioned, we've just started somewhat recently, but in the past year, we've caught a lot of animals. We've put 104 acoustic tags in 8 different species, but we've caught 11 different species. There's over 50 in Port Phillip Bay. But the ones that we've acoustically tagged include the fiddler rays. So we've got 37 females, 21 males, but we've caught almost 300 of them and only had 3 recaps so far. Everything we catch, we also take blood and muscle and genetic samples. We're obviously working with Flinders and Lauren Meyer doing stable isotopes and fatty acids. But we are very open to collaboration, and if there's anything else we can collect while we're doing this, let me know. We have tagged 18 Port Jackson sharks, 11 of the Melbourne skates. We've caught more of them, but they're massive, so we can't actually tag some of them because we can't get them into our boat. We've tagged 9 of the 7-gills, and again, caught some really big ones. And with these, you'll notice we've mostly caught females, which is interesting. We've caught some Bathytotia brevicaudata, the smooth stingrays. We've only been able to tag 3 because the majority of them are over 1.5 meters disc width. So if anyone has ideas on how to easily handle rays with a barb that big, please talk to me. I'm desperate to tag them. We've got a few gummy sharks. We mostly don't tag them because we know a bit about them. There's a commercial fishery for them, so there's previous work. We've got a few of these swell sharks, and we've caught 1 eagle ray so far, but we're hoping to catch a few more. So again, I did say in my abstract that I would present some results. So this is an almost 2-meter female 7-gill that we caught. And she did some interesting moving around Port Phillip Bay. We tagged her on June 27th last year, and then downloaded our receivers about 1.5 months later. And in that 1.5 months, so 2 months of movement-ish, before I play this, I'll just say the X's are where we have receivers and had no detections, and the yellow dots are our receivers that did have detections. So we thought she did a tour around the bay, but in fact, she went up and down mostly and did not hit the coast. She hung out at the bottom for a bit and then kind of went back and forth. And that back and forth is between 40 and 50 kilometers. And typically, it was 3 days back and forth. I know it's not like I don't have anything to say about conclusions on that, but I promised preliminary results. Those are some of them. If you want to see some preliminary results of the fiddler ray stuff, Louisa has a lot more and cooler results. Go check out her poster. And that's really what I wanted to say, introduce our lab, what we're doing, and invite collaboration. We already have ongoing collaborations with Flinders University and the Southern Shark Ecology Group. We work with the Wadawurrung traditional owners, and we're currently looking at seasonal trends using traditional owners' seasons and westernized seasons and seeing if there's any indication of differences. And we've hosted people from University of Malaya and WWF to come and tag with us and hopefully do some tagging over in Malaysia. So yes, if you would like a sticker from the lab, come and find me later. I'm gonna not take any questions now to catch us up, but please come talk to me. And as the moderator, I'm letting you know we have the group photo. I can already hear the bells outside. Everybody go follow the bells and come and find anyone in my lab to chat about collaboration opportunities. Thanks for being here.
First speaker, Diya. Diya will share research on threatened shark populations around an oceanic island. Diya, the floor is yours. Please go ahead.
Thank you. Good morning, everyone. Thank you so much for being here, and thank you to the organizers of Sharks International 2026 for giving me this opportunity to present this work. My name is Diya, and today I will be presenting some of the work that we've been developing in the Azores Islands, Portugal, where we study movement in juvenile smooth hammerheads, and we focus on essential habitats, or something that we can call is their home. So what is home? Or what would be more terminologically correct for those who study animal behavior, what is a home range or an area that is essential for the animal survival? So there are possibly more ways to estimate home range than there are to skin a cat. Recently, Kraft and colleagues did a review of some of the methods that are commonly used in passive acoustic telemetry to measure space use. I think some of you would be familiar with this. And you know that based on the metric you use, the result can vary considerably. However, home range is not about equations, right? And some of us here who have various home bases would resonate more with this ecological definition of what is a home range, where when we change where we are, our mental maps update based on where we are, right? So if it happens with us, why wouldn't it happen with animals that are constantly moving? So as we can see, home range is, or the area that an animal deems important, just doesn't change over the year or like annually, but also over its lifetime or different life stages. So keeping the temporally dynamic nature of what an essential habitat essentially is, what is home is an area that is important to the animal at any given point in time. And those of us who are privileged enough to study for a long term our species and our field sites can actually capture these transitions that happen in essential habitats ontogenetically. That is my home site for some time in the year. This is the Azores Islands in the North Atlantic. I am based in the island of Fayal, which is outlined here in red, which is part of a twin island system along with Pico, which is separated by a very narrow channel, which is about 4 kilometers wide, and not very deep by our standards, only 200 meters. As is common with small islands, the water becomes a natural extension of the land, and the locals obviously use the marine area just as intensively as they do the land area, which means that sightings or the knowledge that juvenile hammerheads are present in these waters was actually pretty common. So much so that in 2010, people from the Department of the Azores and some of my bosses started tagging juvenile smooth hammerheads to see their space use around the island. And the results of this study, the preliminary pilot study, showed that juvenile smooth hammerheads are not just present around Fayal and Pico, they're resident over multiple years, but also they seem to be using a certain area of the north coast of Fayal disproportionately, which obviously warranted further investigation because the limits of the pilot study was that they had only been able to tag sharks that were more than 1 meter long. So we weren't capturing the younger size classes, which made it necessary to look at non-invasive study methods such as BRUVs. And sure enough, we found baby sharks that corresponded to sizes that are what is normally known for young of the year hammerhead sharks, juvenile small hammerheads. So the study resulted in this, what we found in this study with the BRUVs is that the younger sharks seem to be using a more restricted area within what we designated as a larger nursery. And the space use of smaller and larger juvenile hammerheads is not just separated along the island shelf, but also vertically. So the smaller hammerheads are in the bottom, and the larger hammerheads are exclusively using the pelagic zone. So we accompanied the BRIV study with also a very intensive tag and release study with the focus on capturing younger sharks, and found one of our scientific longlines actually seemed to have dropped on a young smooth hammerhead aggregation. And we were also lucky enough to capture this, who is a little guy. He's 50 centimeters long. It was amazing. And we managed to tag this individual as well. So the extension of the study objectives also was accompanied by an extension of the acoustic network. So we tried to cover as much as the coast of Fayal as possible, but also focusing on the neighboring island where some of the local resource users had mentioned that they have sightings of juvenile hammerheads, and also some of the local, the neighboring seamounts. So these are the preliminary results of the passive acoustic study since 2019. The circles that you see here are detections per day at each receiver, but standardized by the amount of time that the receiver has been deployed. So it highlights areas that are being used disproportionately compared to the amount of time that they've been in the water. And it's pretty clear that this area in the northwest, it's a shallow sandy bay called Praia do Norte, is being used a lot more than would be expected for the amount of time that it has been in the water. And we saw that the detections from the youngest individual that we've captured, was almost exclusively, 99%, within this small bay. Interestingly, this other area, which is just about 5 kilometers from the previous site, had no detections of younger individuals, and the probability of an individual being detected there actually increased after it had attained about 3 to 4 years of age. Yes. And interestingly, also to flip the script on the whole north coast of Fayal is very important. We saw that an individual that we fished and tagged and released in the south, so this particular, it's a smaller bay in the southern eastern coast, spent a considerable amount of time just where it was tagged, and it never went to the north. So you can see here that the distance from the tagging location is 0, so that's the residency, but also at a site that's 60 kilometers away on the east side of Pico, where other things are happening that are completely very interesting, because these stations are, just to clarify, these stations are in the water throughout the year. We go and change them in the same day. And you see that the detections here increase dramatically at the end of the year. So starting mid-October, there seems to be the sharks are coming towards this side of the island. And the seasonal signal is so strong that even this 50-centimeter guy who was only detected for the first couple of months within that small restricted area of Praia do Norte made its journey starting in the second or first week of October and made it up to Galego, which is 60 kilometers away. Unfortunately, was never detected again. So obviously, you might also look at drivers, what is changing seasonally, etc. But my research interest lies more in what this entails for the navigational capabilities of these juvenile smooth hammerheads, because one must ask that how an animal that is continuously moving, A, knows the limits of where it can use the area that is beneficial to it, but also B, when it changes or has this migratory impulse, how it knows where and when to go. So this is more my research focus, and I am trying to explore this further. So if anybody in the audience is interested to chat further, want to collaborate, please feel free to reach out to me during the conference or email me. These are just some of the funders that have helped this work happen over the last multiple years, because this study has been going on for at least 15 odd years. That's the core of the team that helps with this work as well. Thank you so much, and I think we have some time for questions.
Thank you, Diya. Really interesting findings on threatened sharks in island waters. Any questions? Yes, there it is.
Was your tagging site only in the North Bay, or did you have multiple different tagging sites, and did you look at their fidelity to where they were initially tagged at?
So we were initially in the beginning, because there was overwhelming evidence that they were using the north coast of Fayal. Anecdotally, all of the tagging was focused on the north. But then we started looking at different places. So we did try and tag in different places. And this particular example, yes, this one, is from tagging at a different site. So it's on the south coast, and we found that the individual stayed around there. And in fact, some of the individual, it's not a very clear, as we've like, a lot of people have discussed this, the behavior, they do whatever they want. So sometimes they'll like follow a pattern which is really useful. And we did see that this individual and some other individuals that were tagged in the south did stay around there and didn't go to the north. But obviously, with age, that pattern does seem to disappear. Yeah. Anyone else?
We can go with one more question.
Dear, great talk as usual.
Thank you.
Are we aware if anyone's used coastline concavity as an explainer for like predicting abundance? I was just noticing at the top of your map there, the sort of exposed coast, there were many fewer sharks than in the bay. We know Elasmo's like a protected bay for nurseries. Is anyone actually using that? Have you heard of anyone using that as a metric?
Did you mean like coastline complexity and exposure?
Not just complexity, like concavity. Like, is it a bay? Almost like the metric of is it a bay versus is it an exposed headland?
We actually see, so in terms of abundance, it's surprising that the exposed rocky areas have greater abundance of the juvenile sharks, but it's a different size.
Right.
And it is very like size specific. So the sharks don't seem to really get, like the larger sharks starting at 2 to 3 years of age, they go out and they're like more present around the exposed headland than inside. So the inside seems very specifically for like the young of the year and a little bit at 1 year and then very sporadic until like 3 and then not anymore.
Right, good stuff. Thank you.
Thank you so much.
Thank you, Diya. Thank you.
Thank you for your attention.
Okay, next up is Kathryn IS. Kathryn will talk about how seasons and predators affect black-tip sharks in a protected marine park in Mexico. Kathryn, please go ahead.
Is it on? Okay, hi, my name's Kathryn, and I'm going to be talking about the black-tip sharks of Cabo Pulmo National Park. So Cabo Pulmo is located in the Gulf of California. You've got mainland Mexico and Baja California Peninsula. So it's been an MPA since 1995, and this was advocated by the local community. Within 10 years of its creation, fish biomass increased by over 400%. Shark fishing is very common in the area, and there's actually a fishing ban that runs between May and July for 3 months, where it's illegal to catch sharks. In Cabo Pulmo, locals noticed large aggregations of sharks since 2008. So in 2013, we started a terrestrial monitoring program where we actually count sharks from the coastline. The issue with this is that it was really hard to count sharks in real time, and it was really difficult to also identify the species of shark. So in 2019, I came to Mexico to start my PhD, and I added drone aerial surveys. So this is the north side of Cabo Pulmo. It's very sandy habitat, and thankfully, the blacktips kind of hug the coastline, so it was perfect to do the aerial transects. The habitat is also very sandy, so it's very easy to see the sharks. So we do the surveys once every week, and we fly in the morning between 8 and 9, and in the afternoon between 3 and 4. Before every transect, we record environmental factors such as wind speed, wind direction, tidal height, as well as sea surface temperature. And then we fly the drone along the coastline. So this is what the drone sees. We always keep the drone we always keep the coastline at the side of the drone's view, and these are black-tip sharks, so these are Calcarhinus limbatus. We fly at 100-meter altitude, so there's not disturbance with the sharks. We also have a south site. This area is a little bit different, so it's rocky reef and sandy habitat. So it was very difficult to do the transects for the blacktips because sometimes they were in groups a little bit further offshore. So what we do is more like a scan, and then we take images of where we see those blacktip groups. We also do a transect for other species. So this is at a lower altitude, at about 70 meters, and we fly the drone along the coastline. So the species that we've seen include bull shark, lemon shark, and Pacific nurse shark. When we first started the surveys, the maximum number of nurse sharks that we saw was 8, and now we can see up to 40 nurse sharks in this particular area. We also see often neonate lemon sharks, so I'm thinking that potentially this is a nursery area for lemon sharks. So the results. So we actually, back in the day, manually counted all the sharks from the frame grabs, and there was between 1 and 1,229 black tips along the 1.5 kilometer coastline, averaging at 282. And what we found was a very strong winter seasonality. So you can see this graph here. We have the average relative abundance, and here we have sea surface temperature. So when the sea surface temperature is over 25 degrees, we no longer see sharks along the coastline. So we actually managed to tag 26 black tips with acoustic Vemco V16 transmitters. And what we have here is a very simple abacus plot. So we have the names of our receiver stations and then time along the bottom. And what we found was the same. In these gaps here is when the sharks are not detected at our acoustic array. So where do they go? So we managed to tag 8 black tips with spot tags, and we managed to get 4 tracks. So this was our longest track. This was a female that we tagged in April 2019. And then she moved all the way up the Gulf of California, over 900 kilometers, where she was caught on the 1st of July in 2019. So what you can see is that the sharks are actually moving to more productive waters, and there's also been nursery areas described higher up the Gulf. Unfortunately, all the sharks were caught during the seasonal fishing ban, showing that there's not a lot of enforcement on this seasonal ban. So we also had some sharks caught that we tagged with the acoustic transmitters. We had one female here that was caught in Sinaloa, again, during the fishing ban. And then we had our first evidence of a black tip leaving the Gulf, which was this male that was caught off Oaxaca. We also found that the black tips were heavily detected at important seamount areas, which are the stars just there. So why are the sharks so close to shore? So we often see this. It's what I call a sharknado. It's where the sharks swim continuously in a circle. So we know that sharks can use warm, shallow areas for thermoregulation, and we managed to model the abundance with sea surface temperature, wind speed, photo period, and time of day. So what we were seeing is almost double the number of sharks in the afternoon surveys when wind speed was almost 0 knots. So when the water's not been mixed up by, it's been stratified, it's a lot warmer. So I think the sharks are basically seeking out these warm areas. Another reason could be socializing. So what we want to look at is if there's any socializing, social structure in these aggregations, because that would be really cool to find out if they have friends. Another thing that we have in Baja is orcas. So we have this quite famous pod now that's an elasmobranchs specialist pod. We've seen them eating mobula rays, we've seen them eating whale sharks, and we also filmed them eating bull sharks in Cabo Pulmo. In January 2022, we saw this pod up in the north site where we get the black tip aggregations. The week after, when I did the drone surveys, we didn't see a single black tip shark, and we didn't see a single black tip shark for 2 months. The following year, we didn't see them either. So potentially, the orcas are redistributing where these sharks aggregate. Now, we see a lot of the black tips in the south. Like if you remember from before, the south is more like rocky reef habitat, which is a lot harder for orcas to come in and predate on black tips. In the north, it's like sand, so it's like this, like a slope, so it's a lot easier for the orcas to potentially capture the sharks. Another reason could be just less sharks. Like we've already seen that there's quite a lot of fishing of these sharks, so maybe there's just less sharks and that's why they're not in the north, but we'll see. So the project now. So when I was doing my PhD, when I arrived, my Spanish was horrible. So I didn't really have much local involvement. And I realized when I finished my PhD 4 years ago that if I really wanted this project to continue, I needed the involvement of the locals. So I reached out to a group of local women who are actually dive guides, and they were so excited to be part of this project. So now we have an educational aspect to the project. where we go and I teach the local women how to fly the drone and all about the sharks that are literally just off their coastline. Also, all my data and the annual reports we share with CNAMP, and they manage the park. It's very important that this project is continued because Cabo Pulmo has a lot of threats, especially like hotel development and a lot of tourist increments around the park that could threaten the ecosystem. So yeah, thank you for all the donors and the collaborators. And if you have any questions.
Yes, thank you, Kathryn. Do we have any questions? Over there.
Hey, thanks for that. Just wondering if detection range could fluctuate with your wind speed, affecting your actual ability to detect them.
Yeah, so obviously, like, if it's really, really windy, there can be, like, white caps, and it can affect turbidity of the water. So we only flew if it was below 15 knots. So we had that range from 0 to 15 and found that relationship that when it's 15 knots, like, the close to shore is not super calm, but it didn't affect if we could actually see the sharks, especially because in the north it's so sandy. But also any transects where I couldn't see the bottom, we discarded. I didn't count those for the abundance data.
Thank you.
Yeah.
All right. So thank you, Kathryn, and great insight how predators and seasons shape shark behaviors in protected waters. Moving on, I'm happy to introduce Margie H. Schooler. I'm really sorry if I mispronounce your name. And Margie will present on how sharks and predatory fish change with the seasons in the northern Galapagos Islands. Margie, welcome. Over to you.
Thank you. Good morning, and thank you for attending my presentation. So today I will be talking to you about my master's project that I conducted at the Charles Darwin Foundation on the seasonal variation of sharks and predatory fish at Darwin Wolf Islands. So Darwin Wolf Islands are actually famous for their high shark abundance, making them a global hotspot for conservation and shark diving. And in comparison to other no-take and remote marine reserves, Darwin and Wolf harbor the largest shark biomass reported to date. As you might see from the photos, the scalloped hammerhead shark is one of the main characters at this site. And it is also the main attraction of the shark diving industry in Galapagos, with an estimated economic impact of over $14 million every year. While at the same time, this species is also highly vulnerable to bycatch mortality, with one of the lowest post-release survival rates of all sharks. So, in terms of seasonal variation, Galapagos Islands lies at the crossroads of three major ocean currents in the eastern tropical Pacific. And the alternating strength of these currents throughout the year generate two distinct seasons. First, the warm season from January to June, with sea surface temperatures rising above 30 degrees, and then the cold season from July to December, with sea surface temperatures partly dropping below 20 degrees. So keeping this background in mind, our study aimed to reveal how these seasons would reshape predator community at Darwin Wolf Islands, thereby delivering the first warm-cold season comparison of composition, abundance, and biomass, and identifying the species that was significantly driving the change between seasons. So, most of the data that I analyzed was collected by diver-operated video, short DOV, and this technique is especially valuable because it allows us to calculate shark density expressed as biomass per unit area, and this can be used for global comparisons. The team consists of two divers, one of them holding the stereo video set, facing forward, swimming at a constant speed along the rocky reef contour at 20 meters depth, and then the second diver towing a surface marker buoy to record GPS location, which can then later be used to calculate the exact area that was surveyed. So here's a little snippet of one of the videos that was collected at Darwin Island. Using a special software, we can then count and measure each individual on each replicate transect and calculate mean biomass at each site. So here's a first look at our results. Based on the video data, the whole community biomass was substantially larger in the cold season than the warm season, with on average 25 tons per hectare in comparison to 7 tons per hectare. However, it was only the shark biomass that was significantly higher in the cold season. And zooming in, we can see that shark biomass was more than 3 times higher in the cold season. Taking a closer look, we found that scalloped hammerhead sharks make up over 90% of shark biomass in both seasons. That's more than 20 tons per hectare of scalloped hammerhead sharks in the cold season alone. So when we visualize this data in the two-dimensional space, and I'm going to try not to scare you too much with this graph, a clear seasonal trend emerges, where the species driving significant change between seasons, here represented as arrows, show generally a larger biomass during the cold season, here represented as the blue triangles. And again, running statistical tests, we found that the scalloped hammerhead shark was driving over 70% of the difference between seasons. So this strongly supports our previous observation that scalloped hammerhead sharks are the main species driving seasonal difference between warm and cold season at Darwin Wolf. These results have strong implications for management. The strong seasonal pattern of scalloped hammerhead sharks at Darwin Wolf likely reflects their highly migratory nature. And the Eastern Tropical Pacific Marine Corridor is recognized as a migratory pathway for scalloped hammerhead sharks, migrating from productive offshore feeding areas at oceanic island MPAs to inshore reproductive areas in Central and Southern America. And as you can see on the map, these movements often span vast distances and multiple political jurisdictions. So to give you an example, our team tagged a likely pregnant female at Darwin Island to track its movement beyond the reserve's boundaries. And what they found was astonishing. In nearly 7 months that this individual had been tracked, it covered almost 6,000 kilometers, traveling from Darwin Island towards Cocos Island National Park, belonging to offshore Costa Rica, and then further to the continental coast of Panama, before turning back towards the international waters of the Eastern Tropical Pacific. And if you pay closer attention to the timing, you can see that it was actually tagged at Darwin Island at the beginning of the warm season, and it was traveling towards the continental coast during the warm season before turning back towards offshore waters in the cold season. So while first this helps to visualize the decrease in scalloped hammerhead sharks during the warm season, it also raises a significant conservation issue, which is that, for instance, this individual was traveling for more than 40% of the time through unprotected waters. So, to wrap up, Darwin Wolf Islands are a seasonal aggregation site for critically endangered scalloped hammerhead sharks. And we therefore strongly encourage higher enforcement of fishing regulations at Darwin Wolf in the cold season, during the cold season, And to scale up these results, also to strengthen policy management at the national, regional, and international level, we would like to encourage the improvement of fisheries management within the Ecuadorian Economic Exclusive Zone, also support regional collaboration to protect migratory pathways, and lastly, contribute to international conservation frameworks, such as CMS to protect migratory species, CITES to regulate the trade in endangered species, and BB&J to enforce conservation measures of biodiversity beyond national jurisdiction. So, thank you. And this presentation sadly only covered part of the study that we've conducted, and now I've been rushing a bit through it, but I hope if you have further questions, obviously you can come and approach me, ask me now, and have a look at our presentation. Thank you very much, especially to my team, who is also supporting me here, and yeah, hope to be chatting with most of you during the conference further.
Thank you, Margie. And we can go with a one question, if you have. Yes? Over there.
Very cool master thesis. Thank you for the presentation. So the stereo sizing estimates, the error increases quite a lot with distance from the dove, right? Did you account for this, and did you exclude animals from a certain distance? And if yes, doesn't that impact the species composition that you're estimating and the biomass?
Very good question, and it is an issue with the stereo video analysis, and actually I've spent several months on quality control of this data, and the R value in these, to get a bit more technical, in these kind of stereo video measurements is still in development and in discussion, but we have excluded, based on the results of previous papers, the measurements that were off scale, so to say. So there's a value that you can relate to that will tell you how, like the accuracy of the measurement, depending on the calibration of the video set. But this is a whole new chapter and a whole new paper, and I think there will be much more discussion on this hopefully in the future. Thank you.
Thank you, Margie. A great reminder of why protecting places like the Galapagos matters so much. Thank you again. And now I would a very special connection to where we are right now. I invite Chetana Madru to the stage. Chetana will present local research on the wing-headed shark from Bathalangundua Island right here in Sri Lanka. Chetana, please go ahead.
Thank you so much. Hello everyone, welcome to Sri Lanka. My name is Chetana Amadoru. I'm from Blue Resources Trust. So today I'm going to share my insights on wing-head sharks from the Bathalangundua Island. So I will start my presentation with a question. Have you ever seen a wing-head shark before, live or dead? If you do, please. Okay, 1, 2. Okay, very few actually. Okay, I also seen like only dead ones. Okay, if you want to see, there's a dead specimen. 2 dead specimens are in our exhibition in the lobby area. So actually, your answers explain why here, because they had a wing-head shark had a vast distribution, but at present their distribution has restricted to very limited spaces. So according to the current records, wing-head sharks can be found only in 3 locations. So one is in Sri Lanka, in Bathalangundua Island. So according to the literature, it says 80% of the population of wing-head sharks has undergone due to severe fishing pressure. So yeah, no wonder they are restricted to only 3 places now. So this is like a small introduction for the wing-head shark. So they stand out one of the most unique hammerheads among the other hammerheads, because they have like wide, extraordinary heads, which also make them very vulnerable for fishing. And they also found in shallow, turbid coastal areas that also make them more vulnerable for artisanal fishing pressure. These are like critically endangered, and this also like updated very recently, but before that it was endangered. So now I know you all know where Sri Lanka is, because you all are in here in Sri Lanka, and BRT has conducted surveys from 2017 up to now, and we have conducted surveys all across the country, covering more than 120 fish landing sites. So, and interestingly, from whole country, we found wing-head sharks only from one place, that is Bathalangundua area in Sri Lanka. So it's in the northwest coast, And in 2023, the area was declared as an important shark and ray area, emphasizing the urgent need of the conservation importance. So Battalangundu Island. This island is really extraordinary because it's very remote. There's no access for the pure water, and there's like very less facilities. Actually, people have to get the water and the food and everything, the ice, fuel, and everything from the mainland through a boat to the island. And there's a small fishing community, like 30 families are living in there, and all are depend on the fisheries. They have like target fisheries for crabs, bony fishes, rays kind of things. And interestingly, the women in this island plays an important role in fisheries, because starting from the untangling the nets to the processing, they are working on those. So, as I said, winghead sharks have like very extraordinary head. So this makes them more vulnerable for fishing. This is how they get entangled. They mostly get entangled in gill net, especially in 6-inch gill net and 18-inch gill net. And so there's no target fishery for winghead sharks, but because of their head, they get entangled, and they are like, we have like very small population in Battalangundu. And they use OFRP boats. For the people who doesn't know about the OFRP boats, they are like 6-meter long boats, one-day boats going for fishing. So as for BRT, like we needed to know more about the fishing. So we wanted to get to know where these winghead sharks are. So we initiated a community-based GPS tracking program. If I say it simply, we just give the fishers a GPS. Then when they go for fishing, they have to turn it on. And when they come back, they have to turn it off. So for each month, few of us are going to the island and stay there for 5 days. And we collect these GPS tracks, and also we conduct data. And also, not only this area, mainly that if you can see the Sinnagundu just down, the second point down from the top, and that is where we are normally conducting surveys. The other points also we have time to time like going and doing surveys, but we didn't record. We couldn't record any winghead sharks personally, but when we are talking to the fishers, they are always telling like winghead sharks, bamboo sharks, and the black tip reef sharks are the first priority, first 3 sharks they are entangling in their sharks. So the winghead sharks are everywhere. They know, they said it's rare, but it's one of their like top list. Winghead sharks are in their top list. And of course, then we ask like, then where you get them, where you catch them? They always say like, behind the mountain, behind the mountain. So we wonder, where behind the mountain? We don't know, it's just a mountain there. So that's why this came. So from the GPS records that we collected, we created these maps. Actually, when seeing these maps, it's really interesting to see this. This gives lots of information. So actually, we have deployed only 4 GPS for the fishers there, and it was not easy because first we couldn't find like 4 fishermen to give those GPS. Now they are like asking more GPS. We have built that trust and the connection with them now. It's really great to see. And in this map, it shows where the fishing locations are. So they are directly going towards that mountain. That's the seaside, and this is the mountain side. So these tracks are only from 4 boats, and these are tracks collected from few months actually. So I cannot imagine how much fishing pressure this area is subjecting, because there are so many small landing sites, and those all people know where the fishes are. This is like the hotspot for this area. So everyone is coming and catching fish in this location. So when we go deep, we can see the fishing intensity actually. So it shows how much pressure these fishes are putting into this area. So now we got the answer for behind the mountain. Where's this behind the mountain? So this one shows where the wingheads are. So we now know where the winghead habitat. Previously we didn't know. They just said like they were there. Oh yeah, we caught, but really. But now we know where the wingheads are exactly. And this is like a great victory for us because this is a kind of a new innovation for us, and we are really happy to see these maps. And also, these are not just the tracks we collected. We asked our fishers to, like, if you get a winghead shark, please send us, please text us with the photos and the date. So when we get back to the island, we know which date they caught, and we collect those tracks also. So we have like a good collection of tracks now. And most interestingly, the yellow dotted polygon is the Bathalangundu istra, as you can see. But the habitat, according to our maps, it's like bit lower, right? So it shows the habitat is more extensive than previously known. So definitely, we need the expansion of ISTRA, because we cannot say the previous location is wrong, because at that time we didn't have any GPS tracking system or anything. We were based on the fishers' knowledge. We were based on the fishing locations. So that's why we made this Bathalangundu area. That's why we gave this location in the previously for the ISTRA. Now we know. So, and also we don't know what's happening about this area, because we haven't done any surveys this area. So we need, definitely we need like more collection of data, more surveys to identify and to identify these critical habitats. And as I said, definitely we need extra in expansion. And also maybe you will wonder, like, because this is like very unique species, maybe it's nationally protected. But it's not nationally protected. But they are like critically endangered, and they are in grave danger. And one more thing, like very interesting, is like when I talk with fishers, they don't know about this species, the uniqueness of this species. It is just a hammerhead for them. Then I told like the reason about this fish. Then they are like, okay, then we need to protect. Then I was like, yeah, you need to protect because you are the community in there, so you got to protect this. If it is not, we will fail as a nation. And of course, we need to protect this species before this species is going to vanish from our waters like sawfishes. And I need to say finally, these, except one photo in here, other all photos, winghead photos I used are from the fishers they sent us. Thank you.
Thank you, Sheshna. It's wonderful to see Sri Lankan research showcase on home ground, a truly meaningful contribution. And with the time, next, please welcome Eileen Luker. Edy, please go ahead.
All right. Okay, let's fly to Indonesia. Let's fly to Raja Ampat, Eastern Indonesia. And from whale shark, let's move on to walking shark. Okay, understanding the behavior and movement ecology of threatened species is important for to develop conservation strategies. However, data are lacking on many small-bodied reef sharks, especially those in remote habitat like in Indonesia. And most acoustic telemetry studies have focused on large, wide-ranging species, leaving smaller benthic sharks understudied. The Raja Ampat epaulet shark is listed as near threatened. It's fully protected in Indonesian waters along with 5 other species in 2023. And it's a benthic shallow shark species, specialist and nocturnal mesopredator. It can be found in 3 different ecosystems: seagrass, coral reefs, and mangroves. And it's a micro-endemic to Raja Ampat and nearby areas in the bird-set seascape in eastern Indonesia. And our population surveys have identified over 1,000 individuals from only 6 sites in central Raja Ampat. And this population surveys indicated a strong site fidelity and limited dispersal and no exchange between location of this study area. So, although the Raja Ampat epaulet shark is legally protected and the population is generally understood, but no data on the residency pattern, on the habitat use, and the deal visitation pattern. So this knowledge gap limits our ability to design evidence-based conservation measures. And to address this gap, our study have three aims by characterizing the residency pattern and horizontal movement of these species, by examining the DL and vegetation pattern in the area, especially in Raja Ampat, and investigating the habitat use of this species. Together, this aim will identify critical habitat and inform localized management measures. So the study area is in Arborek Island. It's a small island in north-central Raja Ampat. It's only, the size is about 10 hectares, and the area, the island and the reef, it's about 100 football fields. So it's a small area, but this area is a manta village, but also home to at least 700 individuals of epaulet sharks, which is a lot, more than the human population on the island, actually. And this island contains 3 adjacent coastal ecosystems, mangrove, seagrass, and coral reefs, and which is make it ideal natural laboratory. So for tagging, we use P13 acoustic tag, so quite small for the animals, and we tag 43 sharks over the 2 tagging period in late 2023 and early 2024. So the acoustic transmitter were surgically implanted into the body cavity of the sharks, and we also collected the size, the weight, and photo ID, and also tagged the shark using RFID. And the tracking period spanned about 25 months, so just over 2 years. So we have 28 receivers deployed across different habitats around the island, and a range test confirmed a detection range of only 45 meters, which is quite narrow. So because of the limitation, we didn't have enough receivers, so we implemented 4 different configuration of the receivers to maximize the data yield. So we strategically place the receivers in the high detection area and relocated one with low detection rates. And for the analysis, we analyzed the residency events and also movement events using VTrack package. And for the network analysis, we use iGraph to identify if there's a the movement network or the clustering within the receiver array. And this is a summary of the tracking results. So most of the sharks we tagged dominated by females and mature individuals. All sharks were detected except for one that was sadly predated by a black tip shark during the release. And we had more than 400,000 detections. And for the short-term deployment from October 2023 to February 2024, we can see that some individuals, they are detected nearly continuously within weeks and months, which showing that that means that the sharks show high residency and also site fidelity. But some individuals, they only intermittently detected by the receivers. And the detection pattern is only concentrated within 2 receivers. So that means that the sharks showed restricted habitat use on the island. For the long-term deployment over 1 year, we observe a clear divergence in residency patterns. So some sharks, they showed year-round detection. That means that they have a long-term residency on the island, but some display only seasonal detection, and some only detected at the beginning or in the midway through the post-tagging. And many individuals were detected by all receivers. So that means that the sharks, they have a broader spatial use on the island. And for the movement, we can see that none of the sharks cross to the adjacent patch reef, which is the closest to the island, is only about 200 meters and separated by 25 meters deep water. And this indicates that the sharks, they have a strong site attachment to Arborek Island. And we can see some connectivity between the north and south receivers on the island, and connectivity between the receivers that we deployed on the reef slope and the flat areas. And similarly, for the long-term deployment, we can still see some connectivity between reef slope and the flat areas. But as we expected, the longer tracking period, more movement and more connectivity were observed. And for the network analysis, it revealed a very weak modularity with only 0.08 of value. So it means that the receivers are not clustering and into a distinct spatial or functional group, and they form a continuous or well-mixed array. And for the DL pattern, this is interesting. We found that there's a habitat shift, a DL habitat shift. So higher detection rates we found on receivers that we deployed on the reef slope, but during the day. And during the night, we found higher detection rates on the receivers we deployed on the flat areas, mangroves, and seagrass. So it's clear that DL habitat shift from daytime sheltering reef slope to nighttime foraging in the shallow coastal ecosystem. So moving forward, the research should focus on 4 areas. The first is to use acoustic tag equipped with sensors like temperature, depth, and activity to better understand what their behavior, and to use biologgers, especially to cover or to understand their movement beyond the detection range of the receiver, and also to examine the environmental factors affecting their movement and residency. And finally, to use innovative fine-scale positioning to track the fine-scale movement of the sharks. And what are the takeaways? So Arborak Island, the small island in central Raja Ampat, it has shark population over 700 and still counting. It's an important area. The whole island, the whole coastal ecosystem on the island is important for these sharks. And these sharks connected 3 different ecosystems and highly use these habitats. So it is clear that to protect the species, we have to protect all the 3 ecosystems on the island and across Raja Ampat. And I would like to thank all collaborators, donors, and partners, and especially local community in Arborak who supported this study. Thank you very much.
All right, thank you, Edy. A beautifully detailed picture of fine-scale shark movement in one of the world's most biodiverse marine areas. And with the time, actually we're moving to the last presentation now.
Okay.
And for our final presentation of the session, I invite Sophy McCully-Phillips. Sophy will close us out with research on how porbeagle sharks migrate and behave across the Northeast Atlantic. Sophy, the floor is yours.
Thank you. So good afternoon, everybody. I'm here to present some results of a collaborative study, and I'm presenting on behalf of the lead author of this work, David Wrighton, my colleague. And this really is a synthesis of a lot of existing data sets. These data sets are coordinated from CFAS, the Marine Institute, and IFRIMER, and everybody is a co-author on this work here. So this is a bit of an outline of the presentation, so why we're looking at porbeagle shark. I'm going to run through some quick migration tracks, talk about the vertical behavior by season, their association to fronts and the seasonal effects of that, and why this knowledge is useful. So for those of you that aren't familiar with a porbeagle shark, they're found in temperate waters of the North Atlantic and also throughout the Southern Hemisphere. But I'm just going to focus here on the Northeast Atlantic, so that is a separate stock. And they were subject to an intense target fishery, which started in about the 1930s north of the British Isles. It then expanded southwards, and they're a high-valued species, so really couldn't sustain this level of exploitation, and therefore, unsurprisingly, as this landing graph shows, the landings dropped off. Yet, it wasn't until 2008 that any form of total allowable catch was brought in. But it wasn't actually restrictive. So just 2 years later, this was reduced to a 0 TAC, and unsurprisingly, poor beagle had to be prohibited 5 years later. And this is where they remain today. So they remain a prohibited species, but actually they're still caught in quite large numbers as bycatch seasonally. And we have some sort of limited knowledge really about the spatiotemporal movements, but also the drivers to those movements. So as I said, this is a large collaborative data set, and this brings together data from 4 different areas, and you can see them in the map here. 4 different sort of release areas, and here are some of the statistics very briefly. So 44 tags were deployed across these 4 different areas between about 2008 and 2016, and it covered quite the length distribution, so from juveniles all the way to mature adults, with a slight bias towards females. And 40 tags had data recovered from them, giving us a 90% data recovery rate. And this resulted in estimated positions of over 8,000 days at liberty. 2 tags were excluded from this analysis because they only had data of 1 week or less. And also 5 tags were physically recovered. So the archival data was actually added to the transmitted data in these cases. And there were about 3,000 days of data that were lost, either due to premature detachment or reporting failures. So here are the maps of the 3 larger areas where more than 1 tag was deployed. And you can see the males are shown in the triangles and females in the circles here. And I just want to highlight some of the larger scale movements out past the Mid-Atlantic Ridge from individuals that were tagged to the west of Ireland, and some more northerly movements up here from individuals tagged in the Bay of Biscay. And what happens is when you actually look at all of these in combination, what we're seeing is that the migration paths from sharks released at different release locations are actually really broadly similar. They're all moving north in the summer, and they're all moving south in the winter. And they're using the shelf edge as a migratory highway, despite taking some of these longer distance excursions. So this message is reinforced when we look at this density map. Each hexagon represents where there are 5 or more occurrences of a poor beagle reported in that hexagon, with the warmer colours indicating a higher density. And you can see that the density is higher around the shelf edge here of the Celtic Sea and the Bay of Biscay. Now, this will be influenced to a degree due to some sort of sampling bias. But yeah, this is the hotspot area that they are using. So when we look at these same data by season, the density plots really indicate a clear inshore and offshore movement seasonally. So the poor beagles are associating on shelf with water of the warmer months. So these are quarters 3 and 4. And they're moving off shelf when the waters are cooler in quarters 1 and 2. So what is happening with the vertical movements during these seasonal migrations? Well, this left-hand box plot here summarizes all of the transmitted depth data. So it gives a point to each daily mean by month. So each one of these points is a daily mean. And it shows that the poor beagles averaging depths of around 200 meters during the cooler months, and this shallows to about less than 50 meters during the warmer months. And one example data set on the right-hand side from the winter, it summarizes a week of data, so giving 1 point per hour over a course of a day. And it shows that the poor beagles are foraging deep, with deep dives being undertaken for most of the day, averaging around 250 meters across quite a narrow range of temperatures, given the weak stratification during the cooler months. However, they're coming to the surface for around about 8 hours during the night and darkness. So in the summer, this is flipped, so the plot remains the same. And an example of the vertical depth data summarizes, again, a week of data from the summer on the right-hand side here. And this shows that the poor beagles are foraging across a shallow thermocline when the water is strongly stratified, and they're undertaking shallower dives, obviously limited by the availability of depth whilst on the shelf as well. And they're doing this for about 12 hours of daylight, again, remaining very shallow in the top 50 meters during the night. So if we take an example data set and looking at a dive profile from a track of over 450 days in duration, and this seasonal behaviour is clearly evidenced here. So the bars at the top, along here, correspond with those shown in the track here. So we have our cooler quarters in blue and our warmer quarters in the orange colours. And you can see there's two clear periods of deep diving behaviour, here and here. And the poor beagles, and these are when the poor beagles are moving off the shelf and they're making deep dives and excursions into the Atlantic where they're diving down to beyond 1,000 metres. While the shallower diving behaviours are mostly in the top 100 metres, and these are seen when the sea surface temperatures are at their warmest. So these movement data were then used in conjunction with information on the position of highly productive fronts to investigate any relationship between the migration and productivity. And what we calculated was that the distance to the nearest front was statistically significant. So poor beagles were going to be closer to the nearest front than if they moved randomly, so evidencing a causal link to their movements. They are actually tracking the fronts, linking the shark movements to that productivity. So taking that same 450-day track, as we saw on the previous slide, and looking at it from a weekly average point of view. So we have offshore here, and we have onshore here. And the left shows there's two types of offshore behaviour, and they are on a different y-axis, just to point that out. But what it shows is that when the fronts break down during the cooler months, the poor beagles are moving deeper. And conversely, in the summer, when the water is strongly stratified, the poor beagles are staying shallower. And keeping with that same 450-day track, this figure demonstrates the narrow range of temperatures experienced during that winter deep diving behaviour when the fronts are weak here on the left, and conversely the wide temperature range experienced when the poor beagles are foraging across this shallow thermocline when there are these productive fronts which we're seeing seasonally. So just to summarise, this is a large collaborative data set, and we really hope it gives a holistic picture of the sort of behavioural ecology and some sort of level of population level inference behaviours here. We found a clear causal link to poor beagle movements both horizontally and vertically when they exploit the more productive fronts, and they're targeting productive environments throughout the year. And this association to fronts results in an inshore and an offshore movement, despite these excursions and larger migrations. And when this is happening, they are moving into waters beyond national jurisdiction, which is an important factor to consider in any future management of this oceanic stock. And during this migration, the shelf edge is consistently being used as a migratory corridor. So even when poor beagles are tagged at different locations, the timings are still matching as they're moving up and down the shelf edge. And this can really help in any supporting of any spatial temporal management considerations. And as I said, this is a seasonally bicor stock, so actually having a good understanding of these population level behaviours can help us understand the bycatch risk that they're experiencing. Thank you.
Thank you so much, Sophie. A wonderful way to close the session with some impressive transatlantic migration science. That brings us...
She will be talking about combining drone and in-water morphometrics to create 3D models and volumetric estimates of whale sharks.
Bring this a bit down. Thank you so much for introducing me. Thank you so much for listening to me and for this awesome event. Very, very pleased to be here. And I'm talking today about one of my chapters of my PhD, and breaking this really complicated title down, I am talking about 3D models that I created to look at body mass, volume, and condition in the end. So big thanks to my supervisors and the collaboration with the Digital Life Lab. So very, very brief, am I overweight? So everyone has heard probably something about whale sharks already. So the largest fish, filter feeding, have a very long life, slow growth, are late mature, and very challenging to monitor because they're migratory, vulnerable in Australia, where I do my research, endangered in the rest of the world. Morphometrics and body condition, mostly with other marine megafauna investigated, is a very good proxy for health. So that's why I'm investigating this. So far, it's been very simple. So we have a small shark, we have a big shark, we have a chunky shark, and we have a thin shark. And the development of drones give us a complete new window into the ocean. So this is just from the day before yesterday. So I came back from the field from Ningaloo yesterday. This is a very beautiful whale shark sucking up all the krill, very directional. So now to some goals of my first papers. I wanted to create accurate 3D models. For that, I use dorsal images, which come from drone footage, and in-water images, which come from diver-operated video systems. And they are already scaled, and that's a validated method to measure animals, as you probably have heard already. And in the end, to create this very precise reconstructions. The second goal was to validate the drone measurements. So to see actually, does the drone measure similar or the same things like the doves do? The third goal was to compare the 2D and 3D metrics by segments. So we basically sliced up the whale shark in 20 different increments, and we looked at the 2D metrics, which means the dorsal width and the lateral height, but also the whole volumetric increments from the model. The fourth goal was to estimate body mass from length and from volume. So there's a known length-mass relationship for whale sharks, and we incorporated volume into this equation. The fifth and last goal was to develop a relative body condition index. So done a lot with marine mammals. Now with whale sharks, we created of all of these models an average body shape. So it's basically the mean geometry, a model that sits in between all of the other ones. So the study site is Ningaloo Reef in Western Australia, known aggregation site for whale sharks. That is part of the drone data collection. So the first year I've flown a DJI Mini 4 Pro, a super small little toy-like drone. This year I've flown a Mavic 4 Pro, both really useful and great. It's a bit of a hustle on the boat with a drone sometimes. So I had like several heart attacks, but they both survived. That's how the data looks like in the end. There's a reference pole, a swimmer swimming with 1 meter reference pole. Of course, altimeter and drones could also be an option to scale the size of the whale shark here in action. And at the same time, we have another boat with some divers using these diver-operated stereo video systems called doves. They're pre-calibrated in a pool environment and clapped by, synchronized by clapping. And there's a person, a swimmer, a friend of mine, and she took the in-water data for me, which is a great but very exhausting job. So we have now these 2 amazing images. We outlined it a bit, and we made some landmarks, so the black lines. And what we did now is we combined them both in Blender, which is an open source software. So we have these 2 image planes. We dissected them 90 degrees. And then we did structured box modeling, as it's called. So you have a model and a box, and you draw a lot of lines, and you edge loops called, and then you kind of sculpture the animal according to the 2 planes that you have. So this is a very, very easy, that's probably not true, but this is an easy workflow, and it's reproducible. But also, once you have this mesh, which I created already for a whale shark, you can use this mesh and adjust it in Blender for all of your templates. So if you have a whale shark and you have like a dorsal image or a lateral image, you can put in Blender this 3D model, and you can, with the grab tool, just adjust this model and calculate volume of your whale shark. So we created additional models, 7 so far, different length, different Wait, so coming now to the second goal that we had, we wanted to validate the drone measurements, and we did that. We were able to show that the measurements are very similar, only given the perpendicular dove images were taken straight. So that also shows that the drone images are actually more accurate, because in Blender we were able to move the models exactly in the position the dove image was basically calculated. So the whole 3D model is all scaled after the 1 meter reference pole. Then the third goal that we had is to compare the 2D metrics and the 3D metrics by segments. So the top graph that you see is, the blue one is the drone width, the red one is the lateral height, and the down graph is the volumetric change along the body. And you can see already the dorsal width and the volumetric measurements are pretty similar. And we also showed that width is more strongly correlated with volume. And the best predictor for volume is width and height together. So the 3D model provides the most accurate volume estimate. And then we were looking at where along the body is the whale shark, where is the variability the biggest. So we had a composite whale shark that I already introduced before, that is the average mean body geometry. And we looked at it, individuals according to this known length based composite model, and we found out that behind and in the middle of the pectoral fins, the biggest volumetric change takes place. So now the next 2 slides are very unsexy. I'm very sorry about that. I make it very fast, so please bear with me. So body condition is super important, and for that we need body mass. And the mass that is so far calculated is just calculated from length. But as you probably know, we are all, like a lot of us have the similar height, but we have a different mass. So volume is super important here. So what we did is we took this known formula and we incorporated the volumetric measurements from this composite whale shark that we created. And we created like a baseline factor. So that is the p-cal here. And then you can estimate the mass for each individual shark. And with this mass estimate, the good thing is we can now look at a body condition index. Obviously, it's only a relative index. And the more models we have, it's going to be scaled and changing probably, where we can look at what we would observe, or what we observe of this individual, and what we would think of given the baseline model that we have, so what we would expect. And as you can see, for example, one in the first line, whale shark 22, has an actual volume, like kilograms of 4,300, and it was just expected to have 3,400. So it's actually much chunkier than we would expect from length-only measurements. So the main findings, we did a reproducible 3D modeling workflow. We validated the drone measurements. The drone outperforms the doves. The 3D models are the best to capture volume, and we increased the precision by including volume in all of mass estimates, and we suggested like a relative body condition index for a composite, with a composite model. So, and that should be transferable to other elasmobranch marine giants and whale sharks in other parts of the world. So yeah, you're not overweight at all. If anything, we should check you're not getting too skinny. Thank you so much.
Okay, thank you so much, Laura Marie. I'm glad to hear that your drone survived. I don't think many others can say the same. Okay. Approach for accurate species identification in shark products.
Cool. Hello, everybody. My name is Paddy Burke, and I am here to talk to you about some of the work that we've been doing trying to explore a new technique called rapid elemental profiling. And here we go. So obviously, elasmobranchs are at a disproportionate risk of extinction. Most of that is driven by overfishing and trade. And despite a lot of these new regulations that we're seeing coming into force from CITES or CMS, is that oftentimes we don't actually know what is being traded, because after something's been processed, it's often difficult to tell what it is, and then there's other problems that come along with traceability inside those supply chains as well. So sometimes it's like trying to manage something and we don't fully understand what we're trying to manage. So with that, if I handed you a box of shark fins today, how would you might go about trying to tell what species they are? Two common ways, at least the first one is something with morphometrics, right? You can try to see if there's the size or the shape of the fin, or if there's any sort of coloration that's still apparent, might lead you to the species. Another way, of course, is genetics. There's been a lot of work that's been done, especially in the metabarcoding scene. I know I've seen a couple of the leaders out here in the conference so far. But what we're trying to introduce is a possible third technique to try to expand that conservation toolbox, as there really is never going to be one silver bullet to try to fix any of these issues. And with that, I want to introduce rapid elemental profiling, used through this instrument here called portable X-ray fluorescence. So this is a very unique tool. It's a handheld instrument that can measure concentrations of elements from magnesium to uranium in 2 minutes. And these elemental profiles, for our examples here with the shark fins, they reflect a species' diet, habitat, taxonomy, and physiology, as many other factors as well likely contribute to that. And together, all of these come together to create what we call a unique chemical fingerprint. And by using this chemical fingerprint and running it through statistical and machine learning models, the idea is that hopefully, if possible, we can identify species based off of this fingerprint. And with that, that takes us to the objectives of our study here. And first and foremost, do different shark species have distinct elemental profiles? And two, if they do, can elemental profiles be used to identify shark species? So to do that, we are based here in Australia. I should have said that. We had a total of 104 different fins that were collected. These were collected from commercial fish markets in Sydney, as well as the New South Wales Bather Protection Program, and some other sources as well. And these crossed 6 different species: silkies, blacktips, gummies, short fin makos, gulper sharks, and tiger sharks. Because we are basically doing opportunistic sampling, we have very different sample sizes for each one. But with this study, we took the dorsal fin, just to standardize it across all species, and then we analyzed at the ventral side of that dorsal fin. How it actually works, the instrument we used was something called an Mvmt M series. All this isn't particularly important, but what is important that this instrument actually gives us 2 different kinds of data types, right? One of them is the concentration data, so the percent across about 42 different elements for our work, and then the raw spectra. So these are the raw spectral counts across about 2,000 different energy channels. And just to run you through some of the boring bits really quick, we use these 2 different data types. We applied some QA/QC, basically trying to make sure that the elements that we're seeing are actually matching diagnostic peaks and biologically relevant. We threw some stats at them, permanovas, caps, and simpers to understand which elements are doing what and if there are significant differences. And then we did some classification approaches using caps under supervised classification, and then some random forest models as our machine learning approach, using a class balance approach to account for our uneven sample sizes across the different species. So to get through all of that takes us to our first objective here, elemental differences between species. And what we saw actually visualized here with our cap here in multivariate space, that we did have a significant difference across different species in our studies. The cap visualization again highlights some of that species level structure in the data here. So these 6 different groups here represent different species, and if we actually look into some of those groupings there, I think there's some interesting information that comes out of that. So we can see at the top there's 2 animals grouped together. Those are our blacktip and silky sharks, which in ecology actually do occupy very similar niches themselves. On the right, we have 2 of our more benthic sharks with our gummies and our gulper sharks. And in the bottom left, we actually have makos and tigers actually having a lot of overlap there. So this information was quite interesting for us to tease out, and I think when we look at the simper analysis, again, to try to understand which of these different elements are actually driving this dispersion in multivariate space, we actually identified 3 main ones: magnesium, sulfur, and calcium, which biologically makes sense, as some of the, let's say for calcium, the calcification alone of some of these different species is quite different. Again, some of the deeper water sharks tend to be less calcified than something like our mako shark, and magnesium is also an important biological element for some of these separations. Now, now that we've seen that these sharks do have distinct elemental fingerprints, we wanted to see, can we reliably classify species? Can we chuck them into a machine learning model and actually reliably get information out of it? To do that, we first looked at our CAP cross-validation accuracy. It's basically a leave one out sort of approach. And we got around 90% accuracy again across the two different data types, again, concentration data and spectral data. But interestingly, when we look at our random forest models, I know this is a lot of information I'm throwing at you right now. I'll walk you through it, no dramas. On the far right, you can see the overall accuracy. This is the performance of these models. The darker shade represents our concentration data, and the lighter one is the spectral data. So with that, you see that our concentration data is performing a little bit better. And those very colorful graphs in the middle there, each one of those columns represents a different species. And the one on the left is that concentration data for that species in the model, and the one on the right is the spectral data. And these three stacked on top are called precision, sensitivity, and specificity. If you don't work a lot with machine learning, these are essentially metrics to try to get a gauge of how well your model's doing and where it may or may not be performing better or worse. Yeah? So for us, sensitivity is how well we detect true species. Specificity is how well we avoid false positives. And precision is how reliably our predictions are. So when we look across these different species, we can see some differences here. Some of our gummy sharks and our silky sharks are some of the better performing ones. Possibly, again, because gummy sharks was our highest number of sample sizes. But where we start to see things really fall apart is with our tiger sharks on the far right there. They don't really seem to do well across any bit of the model. Again, likely this is due to our lower sample sizes for this animal, but I believe there's also an ecological impact that would be reflective in this as well. As we know, tiger sharks are quite wide roaming, and also they feed on a very wide-ranging diet, and I think this would be reflected in their elemental fingerprint. So I think it's an area that we need to increase the sample sizes to get probably a more powerful predictor for our models. Now, what does this mean? We see that we did get strong separation across most species. Again, they do seem to have, at least in our case here in New South Wales and Australia, strong elemental fingerprints. And this led to, again, a rather strong classification performance. Being able to detect 90% accuracy across a 6-factor machine learning problem is well above random chance. So it's actually showing quite a strong performance for us. Highlighting again that those tiger sharks are likely pulling down a bit of the metrics of the model, but something that hopefully we can expand a bit more when we get more sample sizes. And that this elemental variation very likely reflects the ecology or the environment that these animals are occurring in. And some future work that we're hoping to explore with some of this is, again, to explore more tissues. We're really interested in exploring things like teeth, skin, liver, and oil. This instrument here is able to work across essentially solids and liquids with lots of caveats there. To broaden the sampling across regions, seasons, and life stages, this technique will likely work in the provenance sector. We've had a bit of a pilot scale of East Coast, West Coast with some smaller sample sizes that are, again, working quite well. And then to combine with some environmental data for added accuracy. I think we have a bit more layers here will help the models to really tease apart some of these questions that we're still seeing hanging a bit. And one thing I would like to expand a bit more on as well is the actual automation of this process. This instrument is actually able to export data via Bluetooth, and I've been able to create a rudimentary app that basically takes the whole process from scanning to QA/QC processing to machine learning modeling in about 3 minutes. So it's quite a universal tool. And to wrap up really quickly, we saw that these shark species do have distinct elemental fingerprints. Our models did better than we expected. And I think we can support this for some fisheries trade monitoring, but of course, more validation work is needed across a lot of different sectors in this application. And with that, thank you.
Thank you so much, Patrick. We have no more time for questions, but that was really interesting. So our next talk is from Anusha Neranjan, who will be presenting on assessing the vulnerability of sharks and rays in Sri Lanka's bottom-set gillnet fisheries using productivity susceptibility analysis.
Hello, everybody. Welcome to Sri Lanka, Pearl of the Indian Ocean. And hope you're enjoying Shark International 2026, aren't you? Okay, cool. So today I'm Neranjan from Blue Research Trust. So today I'm going to present to you about my work on assessing the vulnerability of shark and ray species. Shark and ray in Sri Lanka's bottom set gillnet fisheries using productivity susceptibility analysis, which is data limited tool. I'm not going to show you where the Sri Lanka is. Sri Lanka is here where you are sitting today. So in my country, we have recorded more than 100 of elasmobranch species, which it makes biodiversity hotspot in Sri Lanka. But sad to say, Sri Lanka can be identified as one of major shark fishing nation in this region that contributes largest mobile catch in the world and also the most silky shark capture in the Indian Ocean region. Elasmobranchs are not occasional catch in Sri Lanka. They can be identified as one of major retained bycatch in coastal and offshore fisheries. When it comes to the IUCN Red List assessments, more than 70% of elasmobranchs recorded in Sri Lanka are threatened with global extinctions. Lack of stock assessment studies retains as major limitation for evidence-based fisheries management. But we can't reward waiting for the proposed stock assessment because we already facing species extinctions from Sri Lankan water. Pristis, long-toothed sawfish, is one of great example for that because these species are already been extinct from Sri Lankan water most recently, and we have no more longer records of these species. As conservation scientist of shark and rays, I was worrying so much about that, which are the consequences we are facing on these species biodiversity losses. So I was keep searching what are the possible solutions to overcome these challenges. See what I found. Productivity susceptibility analysis. It is a semi-quantitative stock assessment method that estimates the relative vulnerability of species to fishing gear, that integrate the biological productivity of the species and susceptibility of species to the fishing gear to assess the vulnerability. So this method widely applied in data-limited fisheries in the world. That is more ideal to Sri Lanka because we are facing data gaps in elasmobranch fish. And also, it is quick and cost-effective stock assessment method that can assess multiple species simultaneously in same assessment that provide a strong starting point and for evidence-based fisheries management and future research. This is the basic PSA framework. So first step is the PSA is identifying the fishery and also the study area. I choose my study area as, I choose Sri Lanka as my study area because it is my mother nation, and also selected bottom set gillnet fishery as the target fishery because bottom set gillnet fishery is the one of major fishing gear used in coastal fishery in Sri Lanka and also it is responsible for a larger portion of bycatch of shark and rays. The second step was the selecting productivity and susceptibility attributes, which are the factors affecting the biological productivity and fishery susceptibility. So I have selected 11 productivity attributes and 12 susceptibility attributes for this assessment. So then it went through the data collection. Data collection played a key role in this study because I went to the field and collected field data, and also I checked available existing database locally, globally, and regionally for the possible data collections, and also I did comprehensive literature review for former data collections. And also, after the data collection, data analysis was started that went through ranking each attribute of the species and also calculating the relative vulnerability through the statistical method called PSA equation. And my result was I was able to plot my result in simple way in PSA plot. Here you can see my result of the PSA plot. So in PSA plot, the relative vulnerabilities of this species increase toward the top right part of the plot. You can see each and every mark on this plot represent one species of shark and rays recorded in Sri Lanka. You can see most of the species are located beyond the green line. That means most of the species are highly vulnerable for shark and for bottom-set gillnet fisheries. When we compare among the shark, rays, and chimeras, you can see blue color sharks, red color rays, and green color chimeras. So you can see red color dogs are clustered more towards to the top right part of the plot. That says ray species are more greatly impacted, are very highly vulnerable than sharks and chimeras. Not only that, I could identify 35 extremely vulnerable species for bottom-set gillnet fishery. Among these 35 species, 27 species were threatened with global extinction. That's not a small number. And when it comes to the national protections, I found only one species have the national protections in Sri Lankan regulatory policies. That is only the whale shark. None of these extremely vulnerable 34 other species have no any protections or management measures in Sri Lanka. That highlight the high risk of conservation, and the protection remains critically low. The beauty of PSA is it is not assessing only the vulnerability. Through the data quality assessment method of PSA, that provides highlights of the critical data gaps of species level to prioritize future research and studies. So here you can see none of the species are not they are as categorized high data quality species. Major portion of the species are you can see yellow color that says they are medium data quality species. And also you can see red color species. We have 7 species. They can be identified as extremely poor data quality species for extremely data poor species. This result highlights the importance of species-specific monitoring and research for highly vulnerable species and also highly data poor species. Overall, bottom-set gillnet fishery poses a high significant threat to shark and rays in Sri Lankan water. Yet, national protection is critically insufficient. The result of this PSA highlights the importance of integrated PSA result into national fisheries and conservation policy makings to expand the protections of the most vulnerable species and also strengthen the species level monitoring, reporting, and research priorities. The key message is simple. If you act today, if you don't act today, it means inevitable loss of these valuable species tomorrow. I want to thank all of my supervisors and funders and supporters and my team to make this research successful. And also, thank you very much, all of you, for listening.
Thank you so much, Anush. Thank you for your presentation, and thank you for all your hard work behind the scenes for the conference. We have no more time for questions, so we will move on to the
Thank
next
you very
talk.
much.
Okay, for three-dimensional marine spatial analysis of abiotic covariates.
Hello, and thank you for having me. Today I want to talk about a R package that I've developed, Shark ABC 3D. So we'll be looking through a number of different projects that motivated this package's development to sort of tell the story of how this came to be. And so in a recent paper, I created these maps to show where in the world held the greatest diversity and concentration of sharks and rays. And so we're used to seeing species richness maps like this map that I made here, a 2D representation of our oceans. This is made by taking a species range, overlapping them with grid cells. Nice and simple. The problem. Sharks, rays, and chimeras occupy almost every marine environment, from the surface to over 3,000 meters in depth. But often analyses for this clade are done in 2D, even when species live in completely different slices of the water column. The environmental variables at the sea surface versus the sea floor are going to be different. A long line scraping or a long line hooking at 80 meters and a bottom trawl scraping the sea floor at 400 meters all occur in the same cell in 2D. And so my first 3D project was with Dr. Alifa Hack. And so I was tasked with figuring out the 3D overlap between fisheries and species. And so this is where typical 2D tooling ran out. We have R packages like SF and Terra for 2D analyses, but nothing straightforward for depth-aware operations. And so to show this, we have a 3D recipe. So the first ingredient is the outline of the species range. So for example, the species range from the IUCN Red List. The second ingredient are the depth minimum and maximum values. And then finally, the bathymetry of the study area. And so putting this all together, we can create this 3D raster stack representation of the species range. And so I took those 2D shapes and rasterized them at a set of standard depths. And then the brown, actually I'll show you the next slide here. So this 3D visualization here might show it a little bit better, but each stack there is one raster, and it also takes into account the bathymetry so that the rasters are only present within the water column. And so we repeat the same thing for the fisheries instead. So we take the outline of the fishing ground, take the depth minimum and maximum of the gear types and the bathymetry, and we can also turn that into a raster stack representation of the volume of space that those fisheries operate at. And then we can then overlap those 3D ranges with one another to get not just an area of overlap between species and fisheries, but a volume overlap between the two. And so going back to the Bangladesh case study, and so with this toolkit, we can then compute the volume overlap between each species range and gear type and visualize that data as a cross-section of cell depths. And so the figure on the left here shows the cells that overlap in 2D. Imagine looking at each of those cells from the side view. And so we have not only the area overlap from the map on the right, but you can also see that there's a difference in overlap on the left between the fisheries depths and the species depths that isn't revealed when we just look at it in 2D. And so if we were to take that workflow and represent it in the code from my package, we first have a function for rasterizing or creating that raster stack from the ranges. And then we have the next function here for calculating the volume of overlap between the stacks. And so while we just walked through visually, showing it in code, it looks something like this. The next project I worked on was working with some World Ocean Atlas data. And so this is a map of the world's sea surface temperature, which I'm sure many of you have seen. And we often use this to describe the habitat conditions that sharks and rays exist in. Again, the problem, this only shows the variables at the sea surface rather than the full depth that sharks and rays occupy. And so for this next project, I extracted abiotic factors like temperature and dissolved oxygen for the full 3D range of every shark and ray in the IUCN Red List with available range and depth data. And so we got those values for 57 standard depths from 0 to 1,500 meters. I have the example of the great white shark on the screen here to show the kind of range of those temperatures across its full depth range in addition to its area. And so showing this as the code, or showing this as the functions in my code here. So we have one utility for downloading and loading the World Ocean Atlas data. And then we also have another function here for essentially cropping that environmental raster by a species range, not just by area, but also by depth. And so that gives us this end result here. And so the next and last project I've worked on most recently is working with Global Fishing Watch data. So they have a apparent fishing effort raster that is global and gear attributed and quite high resolution. But again, the problem is that it's flat. All that fisheries effort is represented in just 2D. So my package gives you a way to extend that 2D raster data into 3D. So here I have a figure with the benthic fishery types. So for example, bottom trawling clamped to the bathymetry of the area around Japan and visualized in 3D. And so this workflow in code, we take the Global Fishing Watch API and take that tabular data. We can convert it into a spatial raster. And then we have a function for taking the benthic fisheries effort and bounding it to the seafloor, and also taking the pelagic fisheries at their respective depth ranges. And so while there are some limitations with the publicly available Global Fishing Watch data, so for example, Global Fishing Watch labels both midwater and bottom trawlers as simply trawlers. And in 2D, that doesn't matter so much. But in 3D, they obviously exist in completely different parts of the water column. But the point that I want to land here is that this package is about the infrastructure of the workflow, not a finished claim about any particular fishery. This workflow is separate from the quality of the data that is going in. As Global Fishing Watch's data improves, that new data can drop in straight into the workflow without removing or rewriting downstream code. And so absorb the improvements in data as it comes. So the approach I took with the development of this package is to try to think like a software developer. Some of the core tools that I used include open sourcing the package, using Git, writing functions with clear inputs and outputs, and writing tests that check the functions against known inputs and outputs. This is more work than writing a typical R script, but it really helps with reproducibility, collaboration, and for the next thing I want to talk about. AI. Scary. So to say it bluntly, if you want to use AI, you better have these tools locked in. They're the essential tools for keeping AI from taking over. But yeah, basically using AI responsibly and increasing our accountability with it. So to be clear, I did use Claude Code for the development of this package, but I did not use it for the methodology or interpreting of the results. And so in terms of future directions for this work, I would be really interested in creating species distribution models in 3D, mapping some 3D movement from shark tagging data, looking at spatial protections and management, and integrating some additional data sets like the Copernicus marine data. So I want to thank my collaborators over the years. It's been wonderful to work with all of you. And you can access the code for this package at the QR code there. And then one quick note before we open it up. Alongside this work, I'm actually freelancing as a developer and researcher. So I'm looking for more projects. So if anything I've shown you connects with your work, please come find me after the talk or drop me an email. I'd love to hear what you're working on. Thank you.
Thank you so much, Jay. We have no more time for questions, but thank you. And can I have a round of applause for all the presenters of this session? This was amazing.
Good afternoon, everyone. Thank you for being here today. My name is Nurul Manik from the Fisheries Resource Center of Indonesia at Rekam Nusantara Foundation. So, every day, wedge fish and giant guitar fish are landed at fish markets along the north coast of Central Java in Indonesia. They're critically endangered, listed under CITES Appendix 2, yet we lack data that covers all landing sites, especially species-specific data cache. So in the next minutes, I want to show you how we contribute to improving that, not by sending more scientists to the field, but by building a collaborative monitoring systems that puts data collections into the hands of the people who are already there, like fishers, enumerators, and local communities. And these systems we called ICAN, the Initiative for Collaborative Fisheries Data Collections. So let's start with the crisis. Wedgefish and giant guitarfish are among the most threatened elasmobranch groups in the world. High-value fins, heavy fishing pressures, and populations that are extremely vulnerable to overexploitations. Tracking them should be a priority. But for that, we need data, and that's exactly where the challenge begins. Here are our study area, the north coast of Central Java in Indonesia, one of the most productive and heavily fished coastline in Indonesia. On average, Indonesia's total sharks and rays productions exceeds 100,000 tons per year, making it one of the largest sharks and rays fishing nations in the world. And these regions contribute significantly to that number. In this area, we documented 6 species for wedgefish, such as Rhina ankylostoma, Rhincobatus australiae, Rhincobatus levis, and Rhincobatus springeri, and 2 giant guitarfish, such as Glaucostegus towin and Glaucostegus typus. So, what are we dealing with? These 6 species are critically endangered, and yet fishing pressure continues, largely driven by the fin trade. And most of these commodities enters the catch as bycatch. And here is the monitoring challenge. The north coast of Central Java is a vast coastline with dozens of active landing sites. Covering all of them requires enormous effort, more than conventional monitoring system that realistically can handle. And that combinations is exactly what makes data collections so difficult here. Which brings us to the data gaps. Our landing site coverage is incomplete. Not all sites are monitored. And what happens at smaller, more remote fish markets simply goes unrecorded. Even when the data is collected, it often stops at group level. Enumerators at most ports are still recording sharks and rays as category, not species by species. So wedgefish and giant guitarfish disappears into aggregate. And open access to existing data remains limited. And we don't have a full picture of who is collecting data, where, and what exactly they're recording. That lack of transparency makes it difficult to identify gaps or to build on what others have already done. So, what is the solution? We developed ICAN, the Initiative for Collaborative Fisheries Data Collections. ICAN is an open access platform designed to close these gaps, not by deploying more scientists to every landing site, but by engaging the people who are already there. Fishers, fish collectors, students, local enumerators, and community members can all participate in data collections using the same standardized systems. ICAN is designed to be flexible because landing sites across Indonesia are not all the same. ICAN has 3 schemes. The first one combines paper forms with the ICAN mobile app, useful for sites where enumerators assist fishers with data entry. And the second scheme is fully digital. Enumerators record and upload the data directly through the mobile app at the field. And the third scheme, using the ICAN web database for more structured office-based data entry, and particularly for consolidating records across multiple sites. Each scheme is adapted to a different level of digital capacity and community context. But all these three schemes fit into the same SQL server. And the data doesn't sit in a locked database, but it's publicly accessible. Once the data enters the server, basic analyses are automatically visualized in real time on our public dashboard at perikanan.org. As you can see here, you can see length frequency distribution by species here. But I should note that the dashboard is still under active development. So what you see here is just the beginning. We are continuously working to expand the visualizations and analyzed features available for users. And now let me show you what implementations actually looks like on the ground. Across Indonesia, ikan has been implemented at 31 landing sites, engaging 26 enumerators and 28 volunteers. And this is what data collections actually looks like. A wedge fish being measured at a landing site, the total length recorded, species identified, the data entered directly into the ikan tools, and simply enough for trained enumerators. And that combinations is exactly what a collaborative monitoring means in practice. And ikan has been deployed across multiple regions in Indonesia. And it's not limited to wedge fish and giant guitarfish. Ikan covers a range of commercially important commodities, such as pelagic fish, crabs, reef and demersal fish, and of course, sharks and rays. And each monitored through the same standardized collaborative framework. And now let's zoom into our focus area, the north coast of Central Java. Across 4 landing sites, ikan has recorded 3,710 fishing trips and 15,575 length measurements for wedge fish and giant guitarfish accumulated from 2019 through December 2025. And these numbers represent years of consistent presence at the landing sites. And that continuing is turns snapshots into trends and trends into evidence. And this is what ikan is ultimately about. Every trip recorded, every length measured, every species identified feeds into a growing body of fisheries data that becomes the scientific basis for sustainable management decisions for wedge fish and giant guitarfish. And this is what credible data actually enables. The visualizations you see here, the trip trends, length frequency, and species compositions are all generated from ikan data. And these are just examples. The same data set opens the door to much wider explorations and more complex analysis. And this data supports NDF preparations and stock assessments, and it enables Indonesia to meet its CITES reporting obligations. And perhaps most importantly, it bridges the knowledge that fishers hold about these species with the scientific credibility that policymakers require. And that is the real impact of collaborative monitoring, digital tools, and community participation, generating credible and timely data for policy and conservation. And I would like to thank the SARC's International 2026 Organizing Committee for the opportunity to share these works. And if you are working on similar monitoring challenges in your regions, I would love to connect. That's all my presentation today. Thank you all.
Thank you very much, Nurul. Next, I invite Georgia Tiller, who will be presenting on developing novel blogging methods to examine how diet influences the movement of white sharks. Over to you, Georgia.
Okay, so good afternoon, everyone. I'm going to be talking about digestion in large sharks and how we can quantify feeding events using novel biologging methods. So studying the trophic ecology of sharks is really important because it helps us understand how predators can obtain and use resources in their environment. And a way we can do that is by looking at their feeding ecology. And while there are lots of different methods to do that, today I'm going to focus specifically on a tool we've been developing, which is called ingestion-digestion sensors. And so these sensors, which if you're familiar with tags, are just a modified mini-pat tag, but they describe the feeding frequency, the depth, and duration of ingestion. So these feeding pills, they are ingested by the shark and they're retained in the stomach. And while they're in the stomach, they're able to measure the conductivity and the temperature of the stomach environment, where conductivity is measured as impedance. And in vertebrates, there are two stages of digestion. So we have the chemical stage of digestion and the mechanical stage. And both of these stages have distinct or characterizable variables in these processes that the pill is able to read and detect. And then we're able to interpret digestion or digestion stages from these sensors. So in summary of what I did from laboratory trials, essentially we found that the pill is measuring the motility of the stomach, or that's what's having the greatest change. So if we take that understanding and we apply it to a digestion profile, we can sort of characterize the stage of digestion. So we have the first stage, which is our lag stage, which is a drop followed by a gradual increase. Then we have our mechanical stage of digestion, and this is where the profile starts to become more varied. And then we have the end of digestion, where we have a maintained value. So this here is a digestive profile of a captive grey reef shark. And so we have known feeding events of mullet and pilchard, but I'm going to focus specifically on this one mullet event. So if we zoom in here and look at this, and we take that understanding that I just explained of those digestive stages, you can see that we've got that initial lag stage, followed by the onset of mechanical digestion, where we're getting that variability, and then we're getting an empty stomach where the value is returning. And this aligns with previous work of what we know about digestion, and these times align in terms of we're seeing the greatest change in motility at about 8 hours, and then we're seeing the stomach values return to an empty stomach based on the time it takes for a shark to evacuate its stomach. And so taking that and having those periods to overlay onto digestive events, we're then able to quantify different metrics, so we can establish thresholds like proportion of time above a certain value or the mean standard deviation, and then you can use these metrics to act as predictor variables in machine learning and essentially develop a method to identify predation events. And this is really great because we can then apply it to free-swimming sharks. So we have deployed this on 20 white sharks, and this is an impedance profile or the digestive profile of one of these events. And essentially what we do is we're going to take that understanding from the captive trials and apply that to these free-swimming sharks and identify feeding events for periods previously unmonitored, which is really cool. So if we relate this back to the broader applications, I'm going to use it in a Southern Australian context because that's how we're going to be applying it. We're going to then pair this ingestion-digestion data from the feeding pills with traditional biochemical and telemetry analysis, and then we're able to identify biologically important areas for white sharks, which directly contributes to the species recovery plan and also identifying ISRAs, which is really great and exciting because there's not really been a method, well, this is a really novel method, a new method at looking at identifying feeding events. Thanks.
Thank you very much, Georgia. We have 1 minute for questions.
How do you get the information from the stomach?
Sorry, the what from the stomach?
The information or the data from the stomach.
So the tag is averted. So when the shark decides to spit out the tag, we recover it and then we download it, and then that's how we're able to get that information. But we're working on hopefully making it so that we can transmit the data, and that would be really great because then you wouldn't have to physically recover the tag, which is really helpful for wild deployments. But we're just sort of at that stage of working out exactly what we need from the tag to transmit before we can have that accessible.
Thank you.
Thanks.
Thank you very much, Georgia. Our next presenter is Edward Cardona, presenting on standardizing electric field mapping to advance shark deterrent research and design. Over to you, Edward.
Hi, everyone. Thank you for having me. Let's jump straight to it. So, shark deterrents have multiple applications, both for fisheries and for personal protection. And of the variety of stimuli that have been looked at, the electrical and magnetic stimuli are the ones that have shown the most promise. So we took 4 electrical devices, 2 for fisheries applications. We've got the Shark Guard by Fish Tech Marine, and then we've got the Repellex by Repellex. And we also took 2 Ocean Guardian devices, the Scuba 7 and the Freedom 7, both for personal protection devices. The reason we're curious about this work is when we look at the fields represented in the marketing campaigns by the companies, it often leads a lot to interpretation, where we're seeing these lobes around the electrodes, but with no field voltage gradient. And then if we look at the scale on these figures, it often just shows electrical field strength from low to high. And the science has looked at empirically measuring the fields produced by devices and comparing that to the voltage gradient at which they react, but there is still a lot of work to do in this area of research. The reason we're interested in this is because we need to understand the field characteristics that the sharks are experiencing when they react to the field. But we also want to take that one step further, and we want to try and understand how we account for objects within that field, particularly important for personal protection devices, because we need to use all of this information to help guide the development of future devices. So this is our method. We call it the clothesline system. It's basically a PVC structure with parallel lines on which we attach the device to the top line, and then we move this loop antenna, which is a copper loop antenna, at 10-inch increments across multiple axes, and then we're measuring for 30 seconds at a very high sampling rate, so that then we can get a bunch of data, which we then run through a PCHIP algorithm to interpolate the data between the measurements that we're making, so we can go from a low field resolution to a smoothed out high field resolution. Now, like I said, that's not the only thing that we did. We also tested the field of the Scuba 7 with an insulator. Here we used our trusty sidekick, Bob. Unfortunately, we weren't able to use an intern for this part. A key consideration to this is we didn't include any of the dive gear, nor did we include the dive tank that would be used in a specific use case like this. So that's just something to consider when we're reviewing the results. Speaking of results, let's get started. So, unsurprisingly, the first thing that affected the field size was the electrode separation. Here we've got 2 fisheries devices. On the left-hand side, we've got the Repellex, which has an electrode separation of about 1 meter. And on the right-hand side, we've got the Shark Guard, which has a much lower electrode separation of about 20 centimeters. And we see that the field extends much further away from the device on the left-hand side than it does on the Shark Guard, which the field seems much more constricted to the device. But that's not the only thing that we noticed. Electrode shape also influences the size and the field strength. On the left-hand side here, we've got the Freedom 7, which is an Ocean Guardian device. It's typically used for free divers and spear fishermen. And we see it's got 2 symmetrical electrodes, which create this symmetrical field, versus on the left-hand side, the Scuba 7, which has 1 of those symmetrical electrodes, but it also has this plate, which would normally be attached to the back of the dive tank. And we're seeing this plate yields higher voltage readings than the cylindrical electrodes of the Freedom 7. Now, when we add the insulator into the mix, what we're seeing consistently higher results when we're measuring the field with the insulator present than with the insulator absent. What this means is when we're testing these devices, particularly for personal protection devices, we need to test them in the same context in which they'd be applied by the resource user in the wild. So, to bring it full circle, what we're seeing is all of these devices have a specific context, and which come from their own engineering limitations. For example, for personal protection, what we're looking for is a device that can be a little bit larger, the battery life can be a lot shorter, and for some devices, the electrode separation is going to be dependent on the size of the user. For example, the Scuba 7, it's a backplate and an electrode attached to the ankle. Compared to fisheries applications where fishermen want a compact device, something that's small, which is going to limit the electrode separation, but they also need a lot longer of a battery life, which obviously influences the engineering limitations of these products. That being said, I'd like to thank the organizers of this conference. It's been amazing so far, and I'm really excited to listen to all of your talks. I'd like to also thank all the collaborators on this work, and I'll take any questions if you guys have any. Thank you very much.
The floor is open for any questions.
Hello, very interesting talk, congratulations. I was just wondering, you went over it really quickly, but have you seen how these electrical fields affect the deterring, like what is the deterring nature on sharks, and if it varies by species?
So how does the electrical field influence the reactions of the sharks?
Reactions of the sharks, and if it varies by species?
Yeah, so it definitely does. There's going to be a lot of talks from my colleagues about the influence of electrical deterrence, so I don't want to steal too much of their thunder. But I think the main thing that comes from this is a lot of it, we typically measure the reaction based on a voltage gradient, but the voltage gradient assumes that the field is homogeneous. Whereas these graphs prove that the field that's produced by these devices isn't necessarily homogeneous, so we need to take a slightly different approach to really understand the true voltage gradient that these animals are reacting to, so we can kind of target that and develop future devices that will be a bit more efficient.
Thank you very much, Edward. Now I would like to invite Martina Arcioni, who will be presenting on validating fecal DNA metabarcoding for elasmobranch dietary studies. Over to you, Martina.
Hi, everyone. Today I'm going to share some insights from an innovative technique to study elasmobranch diets. I'm going to start strong with a little graphic picture. Just to underline how traditional methods are often requiring the sacrificing of specimens in order to assess their stomach content. But what are the alternatives? The protagonist of this speech is fecal DNA. Fecal DNA was used for decades in terrestrial studies, but was first proposed for elasmobranchs only in 2021 by Van Zinnik-Berman and colleagues. You can collect fecal DNA by swabbing the cloaca of sharks. The approach has many advantages. It's non-lethal, so you can grab your swab and then release the animal. It provides a high taxonomic resolution, and it's also very practical in the field. It's quick, it's easy, and it's relatively non-invasive. But the technique still misses a comprehensive validation of the method. The first validation came actually just a month ago by Rayburn and colleagues, who compared the fecal DNA and the stomach DNA from individuals caught in long lines and gill nets. But what we thought is that the context in which this technique is used is actually quite significant. Indeed, to validate the method, we sampled individuals coming from trawling, and we took samples from the cloaca and from the stomachs of the same individuals. Our results show that some of the fecal DNA does actually provide a reflection of what is found in the stomach, but that's not the case for all the individuals analyzed. As you can see in the graph, for the majority of the skates, non-shared taxa were found among the two methods. Plotting the data into an ordination plot, you can see how the two methods completely separate. You have the guts on the left and the fecal swabs on the right, and there is no overlap whatsoever. It's interesting to see instead how the cloacal swabs completely overlap with the environmental controls taken on board, which is the white cloud that you can see on the graph. So my take-at-home message is one of caution. The technique is very promising and it has many advantages, but if you're planning to use it, be mindful of the context you're using it in, and for sure take environmental controls in order to take this contamination into consideration for your results. And if you're interested in seeing the updated results for this research, please feel free to contact me on ResearchGate, and if not, also during this conference, come and chat up with me. And thank you.
The floor is open for questions.
Oh my God. Hi, Martina. Thank you. That was really interesting. We've employed it as well, and we found a lot of the swabs were empty or clean. Did you have a code brown scale to say where was actual poop on it versus where there wasn't any on it, if that makes sense?
Yes, it does make sense. I actually kept track of the color of the swab once I took it. I did not find a significant correspondence between the DNA in the swab and the color of the swab. Definitely, we had some issues also in the lab, and some samples were presented a lot of host DNA, and we think that that's one of the problems of the technique, that the high concentration of the host DNA might inhibit the amplification of the rest of the prey DNA.
Okay, awesome. Thank you. Very interesting.
We can take up one more question.
Hi, that was really cool. I wanted to know what primer set you use and like
if there was anything you did to like try to get rid of non-target DNA, such as like the shark itself.
Obviously on board we took precautions, so you just wipe clean the cloaca before you swab it. And then during the lab phases also you just try to keep everything obviously the cleanest as possible. Afterwards, during data analysis, I removed host DNA from the data set. I did 2 trials before just removing the host from itself, but there was lots of cross-contamination between elasmobranchs, so that was handpicked, you would say, and that's mostly it. But definitely the environmental controls are a huge help in identifying what species in the colloquial swabs might actually be prey and which are impossible.
Thank you very much,
Thank you
Martina.
all.
Our next presenter is Mariana Arguero, who will be presenting on From Aerial View to Energetics, a drone-based approach to studying aggregations of Mabula monkiana in the Gulf of California. Over to you, Mariana.
Thank you so much. Good afternoon, everyone. So, okay, maybe it's going. It's not this one. This is not the presentation.
AV team, we need your support. Her presentation is From Aerial View to Energetics.
Yes. Thank you. So I'm going to present this. It's a part of my PhD, which is a drone-based approach to studying the aggregations of Mabula monkiana in the Gulf of California, Mexico. So Mabula monkiana is the smallest of the 5 mabulas that we can find in the Gulf of California. And something really special about this species is that forms aggregation that goes up to 15,000 individuals. What is not yet known is why they use this aggregation, like the behavior in them, if they use a specific geometry, and if they get some energetic benefits from them. So what we do is to record the entire group using a drone during a couple of hours, and we also take a picture of a live buoy at the flying altitude that we record the mabulas, and with this we will be able to determine lengths and also the size of a pixel in centimeters to later also determine lengths. So first we'll look at what happens within the group. This is by checking the wing beat frequency in different parts of the group. So for example, we can see that the mabulas at the rear had a lower wing beat frequency, so they spend less energy than the mabulas swimming in front. We also are creating etograms using Boris, so to see if there are mabulas changing places, like for example, a mabula moving from the front of the group to the rear. About the group geometry, we take 1 frame every 1,000 frames from a video. And here I got the first picture, but actually the black cross is the center of the picture, which means it's the GPS position of the drone. We manually check the center of the aggregation and the perimeter, and with the information of the flying records and also the pixel size, we can calculate the GPS position of the center of the aggregation, and also we calculate the angle towards the north or from which the mabulas were moving. So with all this information, we can get something like this. The green line is the position of the drone. Just forget it, I was new flying drones. But here you can see from 1 to 9 how the aggregation was actually changing shape and was moving. So with this, we can get the latitude and longitude and analyze how the mabulas were moving. With all this information, we are going to add the surface current from Copernicus to see if there's some relationship with current, because sometimes we see the mabulas swimming in a round shape. In this case, it wasn't like that. So we will be able to see if they have a specific group geometry while swimming towards the current or counter-current, for example. And we already have distance and time. So we already, I didn't put it here, but we already calculate speed. So we are also able to calculate the speed of the mabulas that are swimming and to see also if there's a relationship with current. And that's it. Thank you so much.
We have 2 minutes left for any questions.
Thank you, Mariana. That's a great talk. Do you see any of the mating trends within these large aggregations as well?
Yeah, that's a good question. That's like the first chapter. This is the second of my PhD. Yeah, normally we see the mating trains, separate them from the groups, but sometimes we also see them within them. I'm mating etograms, and most of the time I see the mating trains at the rear of the aggregation, but in the front they start like this initiation, like one mobula chasing another one. So my hypothesis is that they start forming the mating trains in the front of the group where they are more active, and they move towards the rear, form the trains, and they leave the aggregation. But I'm not sure, I'm still checking that, but probably I think yes. Mm-hmm.
We can take one more question.
So that was very interesting. Just perhaps I missed it in your talk. Do they form these aggregations like year-round? When they're in these aggregations, do they do it all day? And does it seem like they're going towards a specific destination, or is the aggregation itself the objective? Yeah,
that's a good question. No, mainly we see the aggregations from March, April to August, but peak it's May and June. Like I'm coming back and going directly to see them, to record them. May and June, which is also the reproduction period. And it's more common to see them early in the morning and in the late afternoon. During the midday, it's really rare. Thank you.
Thank you, Mariana. Now I would like to invite Kaylee Scidmore-Rossing, who will be presenting on examining the effects of magnetic and electrical deterrence on tiger sharks using baited remote underwater video systems. Over to you, Kaylee.
All right. Hi, everybody. My name is Kaylee. I am a graduate student at the University of Hawaii, and I work in the Shark Lab at the Hawaii Institute of Marine Biology. My research looks at how tiger sharks respond to magnetic and electrical deterrence, and ultimately whether these tools actually change their behavior. This project is broken into 3 simple questions. First, why brubs? Brubs are a great tool that let us observe sharks in their natural environment, and they don't interfere with their behavior. Why deterrence? Well, we often think about how we keep people safe from sharks, but what are we doing to keep sharks safe from people? By developing and improving deterrence, we can reduce negative interactions and events like bycatch, which ultimately support conservation. My project tests 2 magnetic devices, a wearable bracelet and a solid-state magnet, and 1 electrical prototype deterrent. And lastly, why tiger sharks? Well, tiger sharks were chosen because of their ecological importance, their prevalence in Hawaii and Fiji, as well as their ability to be uniquely identified. Since January 2023, we've deployed over 1,100 brubs across Hawaii and Fiji, comparing active and controlled deterrence. Footage was then evaluated using 2 programs, Event Measure and Boris, to quantify behavior. We focused on response variables such as bait or deterrent contact, time to first contact, and close pass frequency. Now, my project stands on the pillar of only using first-time interactions of tiger sharks that have been uniquely identified. To date, we have identified 87 unique individuals by using stripe patterns, scars, and other features, making sure that we aren't repeatedly sampling the same shark. While I'm still working with my data, here are some preliminary results. For sharks that came within 3 meters of the setup, brute magnet showed the strongest reduction in contact between active and control trials. Tickler showed a moderate effect, while shark band showed almost no difference between the trials. So right away, we can see that not all deterrents are created equal. All right. This bottom figure shows time to contact. Now this metric was a little more variable overall. Here, tickler again stood out. Sharks took longer to make contact during active trials. The brute was less successful at delaying interactions, but it did show a similar pattern to the tickler. However, it was shark bands that showed the opposite trend, reinforcing that it likely isn't delaying behavior in a meaningful way. Taking a closer look at past behavior, say within 30 to 60 centimeters of the BREV setup, how do tiger sharks respond? Well, tickler and shark bands both reduce the number of close passes. However, brute magnet didn't follow that pattern, suggesting its effect may happen earlier in the shark's approach. Finally, looking at close range, the question becomes, if a shark gets close, does it still make contact? Tickler was the only deterrent that showed was significantly reducing contact at this stage. Brute and shark bands didn't show consistent changes, meaning proximity often still led to interaction. And even though shark bands doesn't show a significant difference, this result is still actually very important. It tells us that this deterrent is not reliably effective at reducing contact behavior. So what does this all actually tell us? Well, we saw that the brute magnet performed the best overall, meaning it was most successful at reducing the chances of sharks making contact. The tickler told a slightly different story. It didn't stop contact altogether, but it did change shark behavior at close ranges, which suggests it may influence how sharks interact in the final moments before contact. Shark bands, however, didn't show much of an effect across any of the metrics that we tested. But the biggest takeaway is this: none of these deterrents fully stopped sharks from making contact. So while we can reduce risk, we can't remove it. The gap between mitigation and true prevention is where future research and innovation still needs to focus. With that, I would like to give a huge shout out to my entire team. Without you guys, this would be impossible. And thank you so much to Sharks International for giving me the opportunity to share my research. Thank you.
Thank you, Kaylee. Our next presenter is Paula Abad, who will be presenting on leveraging metabarcoding, underwater cameras, and geospatial technologies to develop an integrated monitoring framework for shallow water elasmobranch. The floor is yours, Paula.
Hello, everyone. My name is Paula, and for the next 3 minutes, I'd like to take you to the shallow waters of the UAE, where we have implemented a multi-method approach to study elasmobranchs, and this has been part of my PhD studies with the University of Murcia and in collaboration with Emirates Nature WWF. So we all know that more than 1/3 of shark rays are threatened with extinction, and is importantly highlighted in shallow coastal habitats in the Gulf, where we've seen that they are critical for these species, and they are also subject to alarming rates of coastal development. My study focuses on the shallow waters of the Sariyat Marine National Park. This is a fully protected no-take park, and it's an exclusive opportunity because it overlaps with an important shark and ray area. This is something rare that doesn't happen often, and it gives us this opportunity to study, develop this methodology that will be efficient for detecting these elasmobranchs, but also to give inputs into their management. So for this, we conducted 4 surveys over 4 seasons. In 2024-2025, we collected environmental DNA sample to detect genetic traces. We deployed baited remote underwater cameras that allows us to see species, but also the relative abundance. And we also did targeted dives in those hotspots that were identified previously by the other methods to confirm and also to analyze the specific ecological interactions. All of this data was georeferenced and was incorporated into georeferencing tools to allow the analysis of the data and also its visualization. Here, quickly, I'll go over some of the results. So we detected 10 elasmobranch species, 8 of them threatened by the IUCN Red List. 2 of them were the ones that allowed the designation of this important shark array area in Saryats. And what is important to show is a comparison and complementarity between methodologies, particularly environmental DNA and underwater cameras. So points to highlight are that overall BRAVs were the tool that allowed us to detect the most number of species, but that environmental DNA has a higher detection rate, meaning we need less samples to detect more species with a detection rate of 60%. So here we can see how they both detected species that the other didn't. That's why they are complementary. And seeing these results allows us to design what is the exact rate of BRAVs versus environmental DNA samples that we need to be the most cost efficient at detecting these species. And we didn't only look at the number of species, but we also looked at how they presented seasonal changes, how they associated to the habitats, and how they were distributed within these areas. So this allows to guide management because it allows us to give recommendations on how to maintain this long-term monitoring, which season is best to spot these species, what management decisions we should make around seasonality, around zoning. And I want to highlight, as some of the colleagues have mentioned in other talks, that because most of the species detected were rays and guitarfishes, sandy habitats are actually extremely sport and they were quite shallow, even up to 1.5 meters depth, and that they were all concentrated in one of the areas of the park in the northeast, highlighting an importance of knowing where these species are found to determine management considerations. So to finalize, I just wanted to mention that this methodology allowed us to develop a framework, like combined methodology, that is cost-effective and that is replicable, and it was very effective to detect just in one shallow water area of this marine national park, 10 elasmobranch species, 8 of them threatened, and the 2 critical important species that allowed the designation of this important area. And with this, I think this is a promising methodology that can be replicated in similar areas in the Gulf. To conclude, I want to thank my mentor at Emirates Nature, Dr. Mateus Molina, and my thesis supervisors, Dr. Yvonne, Simon, and Jose. Thank you very much, everyone.
Thank you very much, Paula. Now I'd like to invite Gabriel Morey, who will be presenting on improved brew 24-hour adapted for scarce and elusive elasmobranch species of the Mediterranean Sea. Over to you, Gabriel.
Good afternoon, everyone. I'm showing the BRAVs developed by the SharkMed and the Save the Med teams adapted to the Balearic Islands scenario. Credit is due mostly to Agustí Torres, the person behind the concept and design of these BRAVs. Well, the Mediterranean is known to be where shark populations have declined most, especially pelagic large-sized species, making fishery-independent research challenging to their low abundance. And here's where our BRAVs, pelagic and benthic, become a novel tool to complement information on sharks and rays. The main features of the pelagic BRAVs are that the system is mounted on a windsurf board, typically anchored at the edge of the insular shelf. It has unlimited power through a solar panel system with a battery that powers a high-sensitivity camera, a timer-controlled light for night recording, live 4G streaming to check for occurrences of targeted species or the correct functioning of the system, over 1 month of HD video recording on SSD memory, a large capacity bait that we replace the bait every 2 days, and navigation safety elements as a red flag, radar reflector, and flashing light. Here you can see the system setup scheme, including details like the electronics stored in waterproof cases, and an extra camera used in some cases for mid-water filming below the thermocline, typically at 80 meters deep. The latest improvements include replacing the washing machine drum with a top access tank for the bait, which simplifies work from the boat, and the addition of a battery housing below the waterline, providing more stability to the BRAF. As for the Mvmt BRAF, you can see it ready to be deployed. The system is mounted on a metal frame, typically anchored between 30 and 120 meters, 48 hours of power autonomy through lithium batteries, 48 hours of high-quality video recording on a GoPro or similar. Long-term surveys are possible by changing batteries and SD cards every 48 hours, and the camera, light, and batteries are inside the metal or plastic housings. Well, the main advantages of these BRAFs are that they very much increase the likelihood of detecting species with low populational densities. The long deployment time allows the bait plume to disperse over great distances and in different directions due to the changes in wind and currents. The extended sampling time units minimize the occurrence of zero-value observations. HD video allows also to record some of aspects as the impact of bycatch. And for instance, we have recorded numerous blue sharks with hooks and lines trailing from their mouths, occasionally with additional lines and netting entangled, or individuals with broken jaws. So finally, we acknowledge the funding support of the Beatrice Ederer Weber Foundation, as well as the Marillas Foundation, through its SARCO project. And here's a very short video showing some of the species recorded. What? Sorry. Why is it not working?
AV team, can we have your support to play?
Yes. You can see both daytime recording and nighttime recording in the end. Well, that's all. Thanks for your attention.
We have 1 minute left for any questions. The floor is open for questions.
That was a great talk. Thank you very much. Oh, yeah. I've got a quick question regarding your data storage. How do you go with storing it on board the thing? Do you have SD cards, or was there an SSD that you install? I'm not sure if I've missed that.
Well, I'm not the one behind the electronics and all the devices, but I think it's SSD cards.
Okay, awesome.
Yeah.
Do you change them during the deployment time at any point, or only at the end when you get the system back?
No, it can store footage for 1 month right now. Yeah,
incredible.
a router and yes,
Awesome.
with a high capacity storage.
Yeah, great. Thank you very much.
You're welcome.
Thank you very much, Gabriel. Last presenter for this session, Isabel Evans, who will be presenting on comparative analysis of software for the photo identification of the white-spotted eagle ray in the Gulf of Mexico and the Northern Caribbean. Over to you, Isabel.
Hi, my name is Izzy, and today I'm going to be presenting my research for my research master's. And it is a comparative analysis of photo ID methods for the photo identification of the white-spotted eagle ray. So these central Atlantic rays are endangered due to, unfortunately, a lot of overfishing, and they're caught a lot as bycatch, so it's really important for us to find as many ways to help monitor this population as possible. And luckily, their persistent natural markings make them a really good candidate for photo identification, and this is a really good, less expensive, and less invasive option than tagging. This paper aims to compare the semi-automated and traditionally used I3S spot, which is, sorry, I3S spot is traditionally used, it's automated, and it requires the user to map out a reference zone and manually outline those as spots. In comparison to this, WildMe or WildBook have a multiple different servers, for example, Flukebook or Sharkbook, which you might have heard of. Manta Matcha is more normally used for manta rays, but it's also able to be used for white-spotted eagle rays. And this AI-assisted automated system has two algorithms, Hotspotter and MUID. For this project, we use images of 64 individuals and their 74 reef sites, both in and out of the water, and these are provided by Mote Marine Laboratory and the Eagle Ray Project in the Mexican Caribbean. In each photo ID tool, images of initial sightings were entered before reef sites were then also processed. And I recorded if the correct match was found, the rank of the correct match, and the time it took, as well as the orientation of the ray in the photo. And additional outcomes to the study included, we created the first protocol for using Manta Matcha for white-spotted eagle rays. And we also provided suggestions that we were going to send to the developers to help improve this tool for further use and smoother user experience. So on average, looking at time cost efficiency, I3S Spot took over double the amount of time as Manta Matcha. So it's a much more efficient system. And the significantly faster automated system allows for reduced user fatigue and reduced risk of human error. However, it does have to be noted that due to the nature of this being an online server, with higher server activity, it does slow down speed significantly. Whilst the high speed of Manta Matcha is really useful, especially for really busy researchers, it does fall behind, unfortunately, in precision. I3S Spot, with their combined, so Mexico photos, let me clarify this, sorry. Mexico photos were from in the water, and Florida photos were from out of the water. I3S Spot overall was much more significantly faster than the others. Hotspotter was significantly the fastest in out-of-water photos, whereas I3S Spot was still faster in in-the-water images. And due to the nature of citizen science really being a driving force behind photo ID, I3S Spot remains the most realistic with citizen science, most relevant and most powerful tool at the moment. In both Manta Matcha algorithms, images arrayed from the side were analysed, and overall we found that these often only presented match suggestions of that were orientation-based, so other images of rays from the side. This resulted in 12.5% correct match rate, which is much lower than images of the animal from the top. On the bottom, you can see two images. So on the left, you can see images of rays of a match pair in the water and then out of the water. The textured background of Hotspotter was picked up a lot in their heat maps. So to see if this impacted match precision, I used match pairs with images only taken from above to limit influence of orientation. MUID showed no difference, whereas Hotspotter showed a significantly higher match precision outside of the water. This suggests that it's picking up background textures for match score generation. So to conclude, that while Manta Matcha provides a streamlined, fast platform which can aid in global collaborative research, I3S Spot currently provides a more accurate tool for this species and is recommended for continued use until Manta Matcha is further trained and development is undertaken. Thank you for listening.
Thank you, Isabel. With that,